கிளாஸ்ல தண்ணி வைங்க. Hello.
Good morning, sir.
Good morning, ma'am. Am I audible?
Yes, sir.
Yeah, thanks. Great.
Good morning, sir.
Good morning, ma'am.
Good morning, sir. I'm Dr. Rajni Mathur. And I welcome you and thank you at the outset for agreeing to be part of this program. Sir, as you know that today is the second day and yesterday we had a good start. We had the inaugural function with the advisor from the AICT, Dr. Kakade, and Dr. Goyal was also there. He delivered his inaugural talk and we had a good session also on manuscript writing. So I'm sure the audience is having a good time and today will be an equally productive day for the participants and a good interactive sessions. I see our chairperson is also here for the day.
Good morning, ma'am. Good
morning. Good morning, good Dr. Geeta. If you agree, sir, then we can formally start the proceedings for the day.
Yeah, I am fine. I'm opened up my PPT also.
Okay.
Okay. Yeah, I'm good. You can start. No problem for me. Yeah.
Okay. So good morning, everyone. And I welcome you as coordinator for the 33rd Quality Improvement Program to the second day of this program. I'm very happy to share with you that the morning session is being chaired by Dr. Geeta Agarwal. Dr. Geeta Agarwal, she is a dean at our university, Delhi Pharmaceutical Sciences and Research University. We are very blessed to have her as a young dean in the university and taking it forward with all her dynamism and enthusiasm and energy. Madam is also very active in innovation and launching products, and she has an illustrious academic career to her credit. Madam, I really welcome you and thank you for agreeing to be part of this session. And I request you to please introduce the speaker of the day. And the moderator today we have served with us is Dr. S. Lata. She is assistant professor in the Department of Pharmacology, and she is also very enthusiastic pharmacology teacher, and she's been actively working with the AICT endorsed programs, especially the QIP programs. So over to you, Geeta ma'am. Please take it forward. Thank you so much.
Thank you, Dr. Rajni Mathur, for your kind words. I am really pleased to welcome all the participants and eminent speakers of this session of second day of 33rd QIP. And it's really my pleasure to introduce Professor Ramanathan. Professor Ramanathan is presently the principal at PSG College of Pharmacy, Coimbatore. Professor Ramanathan has completed his graduation from Annamalai University. He did his master's and doctor's from BHU and his postdoctoral program from University of Bonn, Germany. Then he joined in GSS College of Pharmacy in Department of Pharmacology. Professor Ramanathan is having various awards, including Best Paper Award, Young Scientist Fellowship Award, PP Surya Kumari Prize, IBRO Fellowship for Asia Oceanic Region 2001, Professional Excellence Award 2011. Dr. Ramanathan has also worked as guest scientist at BMBF, University of Bonn, Germany. He has numerous research projects, chapters, patents, research papers to his credit. He has guided numerous MPharm and PhD students. Sir, I am pleased to welcome you at this platform. And today, Professor Ramanathan is going to deliver his talk on significance of omics in pharmaceutical sciences curriculum. I am very hopeful this session is going to be very useful for all the participants. Now over to you, sir, please.
Yeah. Thank you, Dr. Geeta, for your wonderful presentation. I'll also thank Dr. R.K. Goyal, Vice Chancellor of DIPSAR, and Dr. Rajini Mathur for arranging all these events wonderfully. And I'll be also thankful to the Dr. Lata, the moderator of this session. So the slides, other things are visible. Can I proceed? Is it everything is fine?
Yeah, sure.
Yeah, thank ma'am. So today I'll be talking on about the omics.
May I just interrupt you? Have you started your screen share?
Yeah, I started my screen share. 1 minute.
Yes.
Yes, sir, you're good to go. Yes.
Yeah, okay. So I'll be talking on the title Significance of Omics in Pharmaceutical Sciences. What about in our curriculum, how much we studied and what is its essentiality and where we apply these knowledges. So my content of my talk will be, I'll be giving a brief introduction about how it has been come into a line and an overview of different omics. And I'll be talking a little bit about proteomics, which is more relevant to our biologicals and products which we develop. And many times we talk about the biomarkers. So when we go for presentations or when we, even when funding agencies, when they ask for any specialized projects, they will ask for any biomarkers related projects, they usually call for it. So what are these biomarkers and how we can apply these, or as a pharmacist, how can we intervene, or a pharmacologist, how we can do these types of projects? And finally, what are the challenges and opportunity to bring into this curriculum, and as a teacher, how we can do it in a better way? And my concluding my remarks, what we can do or what we can go to the next level. This is my flow of my presentation. And first thing is why we should know about this omics. What are its essential, why we should know? Do we have any advantage by knowing this extra science? Or when included in our curriculum as a teacher, when I teach my student, they'll ask me why I should study these subjects. What are my benefits? That will be the basic questions usually they'll ask it. But fundamentally, if you see this, our Indian system of medicine, I'll take the example of Ayurveda. So we always gone with our omics protocol through intuitions. And if we know the treatment protocol in Ayurvedic system, everybody will have their own dispensing of medicines and prepared by a own Siddha doctors or Ayurvedic doctor. They dispense it in our ancient period. Now it become more technological, but otherwise it is like that only. But now we put on a word called a pharmacogenomics approach, as individualization of therapy or precision of in therapy. So where the pharmacy students or even pharmacology, pharmacy practice students have been more oriented into a precision therapy or individualization of the therapy. Those comes under the category of this omics period. Plus, apart from this, when we have, say, for example, some genetic disorders are there, if we want to, if in the infant stage or in the fetus or in the inborn diseases, if it is all there, if I know a little bit about this biology, then I can very well appreciate, I can predict these dysfunctions, genetical dysfunctions or inborn disease errors in my earlier days. That may be much easier for me to get knowledge on it. And also we can do an analysis on these types of clinical samples and then do what type of genetic modification has been done is in SNPs or whatever it has happened. And that can help us to understand, to elucidate or prevent the future disease progression. And other areas are like gene therapy, which is again, if we know this, we can also have an application part on the gene therapy. I think which is already there in our MPharm pharmacology new PCI syllabus. And biomarker characterization, which is we are not studying much, but I feel that is one of the major concern which we can think it of. Then about the quality assessment. So quality in the sense again in the lifestyle modifications, or even when you have a when you want to have a, so even I remember one of my colleagues when they want to have a child, they have a hereditary of this inborn disease problems. They go and had a check their fetus samples and they found it is not there, then they carried the pregnancy. So we can also have these types of problems which can also be assessed. So as a whole, when we understand these types of what to say omics, we can very well appreciate the way how it goes. So I'll put it in a different way. You keep it as an example. Suppose if you take the physiological function, it is a normal physiological function which is going to occur in a normal individual without any problem. So when the normal physiological function, when I have a stimuli, that is, for example, when the stimulus can be environmental or chemical or toxic or anything it can be, and this stimulus can able to alter my metabolic functions. So automatically produces the metabolic fluxes get altered. And this metabolic functions alterations can lead to my pathology. And this pathology can lead to functional alterations related to the disease. And again, I look for my structural alterations. The structural alteration is nothing but my cell. The cell is nothing but our DNA or RNA or gene. So it goes to repair or recovery, and I'll get back my physiology. So when I want to understand about my normal structural alterations, like gene type of approaches, then automatically I can go into my normal physiological changes. And also these structural alterations can also influence my metabolic functions because of my enzymes or proteins which has been produced by these molecules. So hence it's like a cycle. Suppose if I want to alter my normal physiology, I have to alter my structural functions, structural alterations. And these structural alterations are nothing but our basic molecular substances, and where I can go for the disease preventions or control like that. And if you take here, that's the reason why we talk more about now genetic applications, then antigen related problems, everything we control it here and do the modifications and get my clarity on this area, repair and recovery, even interleukins, receptors, everything can come out on these understanding about my pathways and biomolecular changes. Hence, when I do these process, automatically as a pharmacologist, I'll get back my physiology. So when I have my normal physiology, then automatically I'll have my normal body function without any problem. So everything resolves on my metabolism, disease, and its recovery through your biomolecules. I'll put it in another way in a 3 different sectors. Suppose if you take my biomolecules or structural alterations, the structure I always feel about the fundamental of our body is the cell. So when I want to have my cell function, that is structural alterations, that is the biomolecules, that is nothing but gene expression products, proteins, and its metabolites. So these only regulates my biological system. When to comment, when to not to comment, when to suppress or when not to suppress, everything is controlled by these basic biomolecules. So these biomolecular changes or alterations can be preventable when I have my system is under control. But unfortunately or fortunately, it is not possible, because when we have a biological system, it has been continuously stimulated by any one of these. I put like a special senses. Otherwise, it is also by our environmental stimulus like radiation, stress, UV, or even the toxic chemicals released or drug inductions. So all these chemicals, even the pesticides, pollutants, can able to stimulate my biomolecular changes, which can lead to my SNPs or post-translational modifications that can alter my metabolic functions. So that can lead to my metabolic fluxes changes, and that can reflect my functional alterations. And these functional alteration of my organs can precipitate my disease progression. So that means we have a basic fundamental is, though it is a two separate line, but when I want to have my genetic alterations generally takes place with my stimulus, which results in my functional alterations. And that's the reason I said in Indian system of medicine, I take an example of Ayurveda, where it is individual system based and they give much emphasis to the metabolic function. So generally what happens is they go with the, when a patient go to an Ayurvedic clinic, they check their metabolic rate, how fast it is, or how dull it is, or how moderate it is, or how slow it is. They try to balance their metabolic function through some basic medicines. Once if they attain that basic metabolic functions get cleared off, then they start the treatment to bring back their normal physiological function. But they never address that to the disease. Similarly, what I want to emphasize here is, when you want to study about the omics, try to understand their implications on the changes. So when I have a stimulus, what type of changes it produces, and their changes resulted in what type of physiological functional change. And that functional change can precipitate any diseases, may or may not be. Some people may be carriers, some people may not be a carrier, some people may be, the gene can be recessive, some genes can be very active. So all those things can come into a physiological function that we call as phenotypic. And generally when you have a normalized in more alteration, then go into the genotypic functions. So when you understand these, definitely we can appreciate the disease progression, and we can also try to have some newer drug inventions or newer drug inventions for the disease, which we don't have drugs even 30-40 years before, but now we talk about the very specific drugs because they are able to understand these types of problems. Okay. So now I put in a simplified diagram. It is taken from a published journal, from Omics journal. And this is the entire system. Suppose if you take, I told about the biomolecules, which start from the DNA. From DNA go to RNA, then proteins, and then metabolites. And sometimes these methylations come to about epigenetics or whatever we get in alterations or even post-translational modifications. And according to this, we have different omics like epigenomics, metabolomics, proteomics, transcriptomics, and genomics. So every omics is have a specific function and specific implications in my disease as well as in my genome as well as in the phenomic type. That is very, very important. Suppose if you take, when I have an exposure analysis of nutrition, toxicology, and pharmacology, so definitely I'm going to end up any one of my parameters. Suppose if you take pharmacological actions that can change my transcriptomes, or toxins can able to modify my RNA methodology, or heavy toxins can modify my genomes by methylations or histone acetylations. So every exposure of these toxic chemical substances can able to modify my omics characters. And this omics characters can precipitate into my molecular phenome that can be reflected in my clinical phenomena or clinical outcome, and that we call it as a disease. Suppose my object here is to emphasize by understanding these changes as a pharmacologist, whether I can intervene and I can prevent the progression of these clinical symptoms, thereby I can get into a normalization. And for that, I need some biomarkers to identify by exposing these chemicals, these substances, these diseases, what type of biomarkers are present in the system, and these biomarkers can imply my clinical conditions or not. Once if I able to note that, then I can make it as a clinical phenomenal changes where I can do some modifications. That is the whole interest of the omics. So omics is again what I want to say is this omics are the basic biomolecular changes occurs when we study due to some exposure or environmental conditions or food or drug or toxins that can reflect in any one of the clinical symptoms like obesity or height or whatever it can be, and that can be evaluated by clinical phenomena. And when I want to know this clinical phenomenal changes across these areas, what type of biomarker I can predict or I can look in so that I can have some ideas about this alterations or changes. So next, I given some examples where people are much working on these. Even if you take, we come across the scientists have found out the gene responsible for obesity. They found out the gene responsible for producing breast cancer, or they also found out the nucleotide changes happens in the patient with cardiac problems, or glaucoma. It is one of the most hereditary diseases. So when I study my pedigree of my glaucoma patients, when I know the alterations of my glaucoma genetic function, then I can ask them put into a risk factors, or even autoimmune disorders like your multiple sclerosis or even Duchenne disease. So where I can take out my fetus mitochondrial DNA in the fetal stage and do the analysis for the genetic changes. Even diabetes, people are more talking about the diabetes and other neural disorders. So I have this many applications. If I have some hereditary or genetic implications are there, and if I study my family history, and if I study my through bioinformatics, if I study the gene mapping, then automatically I can understand whether I have any mutations or SNPs or other changes are happening in my system or not. Then that can help the people to have better quality of life. That is another way I want to put it on these benefits of these omics studies. And I'll give brief definitions. I'm not going in detail about all these omics, but I talk a little bit about the proteomics. So epigenomics. So that is, it is a change affect gene expression without altering the base sequences. So we know about the base sequences like base pairs. So these base pairs will never get altered, but the expressions get changed. There are four main factors of epigenetic machinery, which include DNA methylation. That is one of the major many projects they could do it because of methylation of my DNA. There will be a functional alterations can take place. Though I have my gene expression, basic sequences, but I have some problems. Or histone modification, this is one of the basic functionality where we talk about when we do the measurement in the cellular pharmacology or cellular biology, these histones, we take it as one of the like a beta-actin as a housekeeping gene. So histone also being taken as one of the product to show that the histone is stable, it is not been modified. And similarly, microRNA expressions. MicroRNA, now again, many projects have been come out by intervening my microRNA, whether I can alter my gene expression so that the disease progression can be prevented or not. And also the chromatin condensation to the cellular cycle. So when I want to have the total identity, so this epigenomics, when I study, what I can look on is about at the genetic level, what modifications are taken place. Because of modification, there is a expression of my genetic activities has been either overexpressed or underexpressed. But when I take the basic sequence, there will not be any alterations. Only there will be slight modifications on these processes will take place. And because of that, that can have some implications on my disease. And second is transcriptomes. So this transcriptome is derived from the word transcriptase. After the transcription, that is from the DNA to RNA, after this RNA has been formed, what happens is RNA has to be formed into a protein through a ribosomal system. And we have ribosomal RNA, messenger RNA, transfer RNA, and microRNA. And we have other non-coding RNA also there. But when you see this transcriptome, so it is not as simple as even after transcription from the DNA, the RNA has to come out of the nucleus, and in the cytoplasm, it has to get manufactured in the proteins. Or during this process of this transcriptional changes, transcription to a protein conversion, what happens, every stage there is a possibilities that this transcriptional informations can get damaged or altered or truncated, any one of those conditions which the system may not allow it to form the protein to synthesize because of these, any of these one of these issues. And again, if you study in our cellular biology about the pre-transcription or transcriptional and protein formations in between the stages, so we have many issues are there. The protein, all the transcriptional informations will not be converted into a protein molecule. So we have many disturbances can occur, and protein will form only at a low level or can be very, very less. And what could be the reason is because this transcriptional activities, any of these RNAs can get disturbed. And because of these disturbances, what happen, it may not have any proper protein levels. And best thing is any inborn errors in metabolism, generally we have this transcriptome analysis. So base studies have been applied to some of our inborn error metabolism. So when we have a child is born, when it's having metabolic dysfunctions, because it clearly indicates that particular protein or enzymes or targets has not been formed, and that can lead to the damages. So this is another area where we can look in. At least we can study what is these transcriptional proteins and how it can be modified. And many projects now been applied by taking miRNA and silencing RNA as one of the target, especially for the neural disorders where we know about genetic manipulations. So this is another area where I think biologists will appreciate. And if you see the projects, if you go through the many papers, they have studied plenty of RNAs which can intervene or interfere in our normal physiological function. So how to silence those RNAs, or if some RNAs are silenced, how we can activate it. So that is the reason, that's the way how we can get back our physiological function. So as a pharmacologist, I feel there is a paradigm shift is taking place nowadays in our regular drug discovery program also. So if you don't emphasize on these lines as a teacher, at least we should incorporate some of these knowledge to our students so that whoever may be interested, they can pick up these informations and they can start working on it. Or at least they started studying it, and wherever there's a feasibility, they'll start working on this line, which might be better for their benefits. Okay, that is a transcriptome. And metabolomics. This is another major area where, again, when we want to give a biological system, so what happens, we talk about this accumulation of food particles inside the cells, accumulation of proteins, accumulation of lipids inside the cells. So these are all some of these metabolic dysfunctions where the cell will accumulate these types of extra food materials. And these food materials may not be useful to them. That can lead to some damages. It can be either oversynthesized or not been cleared out due to poor metabolism, or it can be any of the other reasons. And because of this, this metabolites, it's nothing but, I'm not meaning any drug metabolites or starch metabolite, glucose. I'm not meaning that. Suppose every cell can synthesize its own products, and these products has to be eliminated from the biological system through either cellular migrations or metabolic functions. Suppose if the system is not able to eliminate, if start accumulating due to some reasons, what happens? These metabolites will become very dangerous to the cell. And this cell will start becoming, if you study in your pathology, I think how many of you food accumulation is one of the major concern for disease progression. So when we study these metabolites, then automatically what happens, we can understand where why the reason is. And this metabolic disturbances is also indicated in many diseases. Best example is steatosis, the alcoholic fatty liver. So now we talk about non-alcoholic fatty livers. So this non-alcoholic fatty livers, again, the accumulation of fatty stuffs, though the people are not taking alcohol. But what is the reason for it? What is how it happens? It's that simple. I'm giving an example. So this is the way how we can know about the disease. Suppose if I read that particular paper, then I may not appreciate what the scientists want to say or what really the people want to convey. That is about the metabolomics and phenomics. So it's a very recent coined word, like phenotypes, one recent coined word. It is how I told about the gene respond to environmental changes. Suppose if somebody is, for best, I can give you a simple logic is about erythropoietin. Suppose if I go to a higher altitude, I want erythropoietin has to be produced much a higher rate to have more erythrocytes production. That means my erythropoietin is a hormone. If that hormone has to be stimulated, that means I want the gene has to be stimulated. Why the gene has to be stimulated? Because of my hypoxic status. The simple logic, hypoxia, because of hypoxic status, the gene responsible for erythropoietin hormone synthesis has been promoted, and because of this more erythropoietin has been synthesized, and because of erythropoietin synthesis, erythrocytes has been produced much higher site that leads to polycythemia vera. A simple, I am telling you an example for environmental changes. Suppose if somebody is having higher altitude, cold environment, higher stress, so how my system, my gene will get respond, and these responses, what happens to my molecular level? Suppose if I expose to my, even I can give you some examples. In the plant system, when I want to grow a particular alkaloid or particular glycosides or proteins, what I'll do is I'll deprive that particular system for the particular plant with the particular component. So automatically there will be overexpression of my gene that can lead to produce more amount of this, sorry, more amount of the alkaloids. Or even if I put under stress or under more sun or more water, then automatically my plant will adjust and produce the chemical which they can prevent the damage from those particular system. So this has been studied most widely in the plant system. Similarly, they want to explore it in those situations. What happens to my environment gene when I expose to environmental changes? What happens to at molecular level? And this is called phenotyping, and it is called phenomic analysis. So because of this, that typing, that is because of genotype, the phenotype will get changed. And because of this phenotype changes, when I do the analysis, that is called phenomic analysis. That is another another area where people are now started working on this to understand about the genetic functions and its link with my environment. Okay. And last about this proteomics, which I'll talk a little bit lengthier, because this is how more relevance to our pharmacy and pharmacology, as well as in pharmaceutics or analysis. Any field you can put in, it have a lot of implications. So proteomics is mostly we talk about with the biotechnology people, and on the biochemistry we talk about the protein synthesis. But what I want to say here is proteins are a biological system. These are all you know very well. And post-translational modifications is the major one what we study in the proteomics. We never study about protein synthesis here. In the proteomics, we talk about post-translational modifications. That is, again, it's a very highly specific, and because of this post-translational modifications, the protein changes its identity and its characters. And because of that, what happens, it can have different functions also. That has been studied most widely in the proteomics. And this proteome can mainly be analyzed using MS, that is the mass spectra, or by protein microarrays, are not been much used routinely in clinical laboratory practice. So this is a simple protein synthesis, just to have a refreshment. So this is a DNA. After DNA is from mRNA, and this is a ribosome. So the ribosomes forms the protein, and this protein will go into a site of action, where it want to have an action, it goes into an action. So from DNA to RNA to protein, it's a normal protein synthesis without any confusions. And I told about this transcriptomics, and this transcriptomics have a major role here. After the transcription to the translation, this mRNA, transfer RNA, microRNA, siRNA can get disturbed here, so that the normal expected protein cannot be formed. So this is the transcriptomes comes here, and proteomics comes after the protein synthesis. What happens to my post-translational modifications? That is the transcriptomics. And genomics comes here, so where my genetic alteration takes place. And because of the genomic translations, what will happen to my entire problems, entire physiological function, that is called as a phenomics. So genomics, transcriptomics, proteomics, then I have a phenomics, and in between, when I have this metabolic disturbances, accumulation inside the cell, and those things are called metabolomics. So all these omics have a definite functions in our biological system, and everything have its own applications in our pharmacy field also. Okay, so this is the transcriptional regulation. So what I want to say is, I told about this degradation. So you can see here, this is a microRNA, and this microRNA is again can be a complementary to my protein, that is the translational protein, that's the mRNA, which can able to synthesize in a different way. But if this microRNA is able to inhibit and cut, they can be able to cut it here itself. There is no need to form a new proteins at all, and thereby it become truncated, and this become degradation, and there is no further system will be formed. But suppose if I have a normal thing, it go into a very easy way translation and forms into a protein structures. And this is the one about the proteomics. Suppose if a protein is formed, the formed protein cannot be as free as we think. It is not as simple as, because this protein is always into a structure. If you see your protein data bank, you can see this type of structures, where I can have this helical structures, I have a ribbon structures, and this ribbon alpha helix and beta helix structures, which is a characteristic feature of the proteins. And why this protein form this structure? Because of the presence of cationic and anionic amino acids, and these cationic and anionic amino acids can form hydrogen bonding, and because of this hydrogen bond formations, it can able to fold into a different way, how it can form into a stable protein considers. Once if it considers a stable form, when it comes into a circulation, there may be every chance that these proteins can be joined with other molecules like ubiquitin, or it can go with a lipid, or again sumo, that is again sumolation is also another type of protein which can able to link in, or it can get phosphorylated, or acetylated, or methylated, or glycosylated, or it can form a disulfide linkage, or can form hydroxylated. So any one of these changes, my protein will undergo, that is unavoidable. And when the protein undergoes these post-translational modifications, it can able to have its characteristic change, because when the protein is attached with some other protein, what happens, it and some other component, it changes configuration. So once the configuration changes, automatically its performance, its characters, everything will get changed. And in those situations, our still many of our pharmacy people, those who are employed in protein analysis by say LCMS, MS, or with MS, they have these problems, because what happens, the original protein is this, but when it comes into a synthetic part, or after the synthesis in a laboratory, or in a say produced in fermentation technology, so what happens, these proteins may also have other translational modifications. So determining these protein changes occurring in the system is a big challenge, and its characterization, and its purity assessment, stability. So everything comes under this proteomics, and again the formulation of these proteins as a drug into a delivery. So you can think it of as a, if I know about my proteomics and most of the drugs like biological or biomolecules or biosimilars, which are all this protein in nature and peptide in nature. So when I want, if I don't know these basic about my proteomics, I will not appreciate how to do analysis, how to do formulations, how to measure its immunogenicity, how to measure its stability. I will not appreciate. So understanding these fundamentals about the protein behavior, protein isolation, characterization, stability, analysis, little bit about at PG level or PhD level will definitely will have an edge because we know about drug well, its mechanisms very well. When you have a little bit about the basics funda, then it might help us much easy way as a pharmacy professionals. Okay. And I give some examples. So what, how many, see, when we studied pharmacology, we never talk about insulin alone, we know much. We know now about vasopressin or desmopressin and all, but now we have all these peptides, even angiotensin peptides. We are not talking much about angiotensin peptides. In 95, 96 only it started much into a big level. So when you take these, you can see here the drugs like for protein drugs, the cancer, metabolic disorders, you can see 18% of drugs are for metabolic disorders. Hematological disorders like hemophilia, blood clotting, immunological responses 16, genetics 9, hormones 13. You can see here it have wide range of applications in diseases, respiratory, neurological, adjunct therapy, eye, cardiac, bone, hormone, genetic, immunological, everything. It has been now started replacing the other drugs where we have more of peptides and more of the other drugs which come into the market. And generally, the protein drugs, I am not going in much detail. You can see about now the vaccines. So now the COVID-19 period, people are not talking about this vaccine production and its formulations, and big companies are coming into picture, and everybody wants to formulate it, study its efficacy and its conditions. And so again, if we know about the basic understanding about my proteomics characteristic features, I can have it. Suppose if I want to have diagnostic kits, many companies have produced, many biotech people, biotech lab have produced kits for COVID-19 test kits. So where they want to develop an antibodies, specific antibodies to an antigen and then test its color. So like this, it have wide range of applications in our pharma field. Apart from this, in therapy, we can have interference with the molecular organisms as well as the proper therapeutic agents, which can replace my that is the, or even your normal, which can be much easier for people to understand. The best is to start with insulin. So insulin is the best known substance where we study for a longer period of time. And to have some ideas, so if you take monoclonal antibodies, we have more than 350 in numbers in the development stage. Vaccines, more than 250 are there. The other biologics, more than 100 are there. And most of the pharma companies now they are shifting into this line. Instead of going for a smaller molecule, they started understanding this structural molecules, structural biomolecules like gene products, proteins, and its metabolites, and understand these either the microorganisms or into the human system, and try to develop an antibody for that so that you can remove it from the system and you can have more specific treatment. That is the way how the trend is slowly changing. And you can see here in another next 2030, you can see here 15-20% of growth in every year is there in this Indian market. And you can see here oncology drugs is the major in the immunotherapy, and antibiotics have 7%. So why I am telling these things is why somebody asked why I should learn this, why I ought to understand these things means these are the potential is there. Suppose if we don't capitalize this now, in another 10 years, if we don't prepare it now, in another 10 years, it may be difficult for us to achieve it. And you can see here 20 to 40 billion is in India alone. That is a big market. If at least let them know as a teacher, you let them know what are these terminologies. And if they know about the terminologies, techniques can be learned later, but at least they know the terminology that emphasize. If I include in somewhere in the curriculum wherever there's a feasibility, so definitely we can have it. And especially I remember in MPharm first semester pharmacology, we have one unit, especially one subunit, especially for, I think 4 subunit is especially for pharmacogenomics and all these areas. But still we can talk it more to our students. And this is about the peptides which are there in current. And you can see here, not many peptides have come. The number of peptides reduced, 2 to 10 has been reduced, but they have number of more amino acids. Peptides more than 50 is less, but the unknown is this many is there. And you can see here, there is a gradually every year, 1899, they started where I completed my B Pharm. And this is where I started my M Pharm. So you can see here the peptides. I remember about the angiotensin peptides and other vasopressin peptides where we studied through the journals and its importance in those period. And now you can see here, now it become into clinical side, another 10, 15 years of time, it can now become more in clinical side. So now if you don't start now on this line, it become very late. Okay, and these are the clinical side, clinical current preclinical condition. These have, we have this many components are there in the current clinicals. So I given all these explanations why we have to study in curriculum. So I given every place, I given examples, I given the emphasize and what are the diseases, and you can answer all these questions if I listen to my lecture. So that's the reason why I say it is very much essential for us to link somewhere in our curriculum, a little bit in depth, where it is possible. We have to include all these aspects, and definitely better than us are our biotechnology people, and we should also know how they are competent. Because when you take chemistry, again, synthetic organic chemistry are people are much preferred in our companies when they go for synthesis. But when we want to designing, they prefer our pharma students. So similarly, when they want to know about this formulation aspects, pharmacological aspects and analytical aspects, I think our students are having much edge than the other discipline, science discipline students, those who are studying. So I little bit few slides, I'll touch upon biomarkers to make it clear in another 5-10 minutes, then I'll wind up my talk. So this biomarkers is a very fascinating word. And still I remember a few years before, we had a very lengthy discussion with our science team in our PSG to have a proper biomarker, because that time, 6-7 years before, we had one project proposal come from DBT looking for a biomarkers for a specific diseases. So we thought we can take about this prostate cancer. Do you remember, I think you remember when you study pharmacology, one of the biomarker is serum prostate antigen. That is one of the Brennan prostate antigen or serum protein antigen. Prostate antigen is one of the biomarker to show or to know that whether that particular patient is having any prostate benign or cancer type. Or even another example I'll tell you about HbA1c is glycosylated hemoglobin. That is again another biomarker. Now the range is 6 to 7 as a biomarker range, where they have it for diabetes. They say whether people are having, same way we have cardiac disease. When you go, we have biomarkers. So every place we have biomarkers to know about this particular informations to help us, the patients or the clinicians to understand, or even scientists to have a data to know whether the particular patient is affected with any disease or not. So a biomarker is defined as a trait that can be objectively measured and evaluated. That is very, very important. So it has to be measurable and should have an evaluation, and it should also have some endpoint, a measurable endpoint that may be used as an indicator. Suppose if I give a drug X, Y, or Z, that biomarker has to get altered or reduced. Then only I can say it is an indicator of a disease or physiological state of an organism. Even I can say BP is one of the basic informations, 120/80. But even after some people say 100 plus age. Even there is a lot of controversies going on whether the cholesterol level is 200 milligram per 100 ml is okay or not. Still, we have a lot of debate is going on. So ideal biomarkers should be highly specific and sensitive. That means a small changes happens in my physiological function. That biomarker has to be expressed well in my body, and that can be a clear indicator. And when I give a drug, the drug or the treatment or any intervention, that has to goes down. That is the way the biomarker has to be defined. And when you take a biomarker, it's one of the big discovery program. If somebody detect a very good biomarker for any good disease, that is path-breaking discovery. I given somebody because that can become a diagnostic tool, that can become clinical tool, that can become a regulatory markers. Anywhere it can fit in, and that to become a guideline to a guideline given by either ICMR or particular people. Suppose if you take in India, then ICMR, so even if you take COVID-19, so now we have when to discharge, what to do, when to a patient has to be admitted, what is his indications, how long the patient has to be treated. Some people say they are asymptomatic but testing positive. Some people say they are not symptomatic, but there is no, the answer is coming negative. What is the reason? Because we are not having any much clarity. That's the reason why we can keep on changing their level of approach, or the protocol guidelines has been keep on changed. So a biomarker, if anybody wants to work it on, it's well and good, but they should keep it and very clear in mind that you should have these 4 basic characters. If it is missing any of these 4 characters, then it may not be a good one, but it can go into research publications, but can become a very, very valid one. That is one which I want to emphasize. And these biomarkers are classified based upon pharmacodynamic way, that is prognostic, or based upon the populations to a responders or non-responders, or another history of disease to call it the clinical indices. That is natural history of disease, that is hypertension, and the clinical conditions. And type 1 biomarker track the effect of intervention as to the drug mechanism of action. Suppose if I intervene the drug with X, Y, or Z, then the particular drug level is get reduced, then can become type 1. But type 2 is again surrogative endpoint that predict clinical benefit. Suppose if I treat these, what will be my benefit out of it? Suppose it can come automatically, gets a clear, okay, the patient is clear, there is no problem at all. So one is direct clinical measurement, second is on intervention of the mechanism of action, and third is the endpoint benefit to the clinical situation. So any of these 3 way, if I can have a biomarker, that is good enough. But again, I have to differentiate that in a patient population, those who are responding and those who are not responding. If I say biomarker X, Y, or Z, I have to show that the patient, those who are affected with X disease, having this biomarker, when I do the treatment dynamically, they responding it well. That is one set of approach. Okay. So as I said, biomarker need to be addressed analytical validity, clinical utility, and regulatory and ethical compliance. So this is the very important. Suppose if I have a biomarker, if I do everything, but if I cannot utilize it clinically, and that cannot become a guideline, then people will say it's okay. So like that, we have plenty of data are there. Suppose if you take creatinine level, it's one of the best indicator for clinical conditions. And if I say creatinine level is less than 1, I'm very comfortable. The kidney is functioning well. The patients can be stopped with the drug. That is good enough. Similarly, when I want to have very more clarity on my system, if I don't have any, if I say a biomarker, keep it on mind. So those who want to do research on biomarker, it is well appreciated, but have it in mind that understanding the basic physiology, and of course the pathology is very, very important, and its expression. If I link with your biomolecular expression, suppose if we take muscular dystrophy, the chromosomal deficit. So if I do the testing in the infant, if I assure it, if I have that particular chromosomal deficit has been measured by XYZ biochemistry, then definitely I can say that the fetus might develop muscular dystrophy at later phase if it is a boy. So better to terminate the pregnancy. Like that, we have every data, we should have a clear indication that can become clinical utilization and can become a guidelines. So that we can, because we are going to play around with the life. So that we have to be very careful and make sure that it is happening very well. So points to be considered is data quality, integrity, reproducibility is very, very important, and sample size. That is again, the people say 500 is okay or 1000 is okay, but still, unless otherwise it is not reproducible, then people will not accept it, and should have clinical relevance, and again should link with the pathological changes and have some therapeutic appropriateness. So when you have all these linked together one by one, one by one, one by one, one by one, then that can become a best biomarker. So I wish some of you can take up this as your future, and you can emphasize on this line and then study how it can work well. So these are some of the techniques which I taken from the Oxford University book. So they talk about, as a pharmacy person, if I want to know, if I want to establish any one of these system in my lab, like see, all these things are chromatography or analytical technique, protein separations, mass spectroscopy or crystallography, NMR. So any one of these areas, if I, even the Western blot, ELISA, anywhere, molecular pharmacology, molecular biology, so anywhere if I fit in my system, if I know, understand to apply through this technique, the proteomics or genomics or metabolomics or transcriptomics, it might be easier for me to learn. So my request is, if any one of the student can take up our lab, analytical lab or pharmacology lab, I'm not put anything about the formulation aspects also here, the protein drug delivery formulations. If any one of these areas, if we can emphasize as a pharmacy professional, which not compete with others, so definitely which might help our students. And I request the teachers, even we emphasize my department people also, to focus on these lines to our students, so that if anybody can take up any one line, if they are very mastered in LCMS, MS, only spectroscopic analysis or only on protein synthesis, that is good enough for them to be a very prominent or promising leader in his field of science. So last two slides about the challenges. So my feel is we have to expertise ourselves to teach these topics. And again, infrastructure to have hands-on training is one of the toughest area, because they're all very costly, because we don't much study about the informatics data analysis, other things. And in curriculum, we have some chapters have been emphasized, but if you want, we can allow them to deep study in depth with industrial collaborations. And those who want to do practical projects, research, it is very expensive, and we need a lot of fund. And again, another major drawback is when you prove it in a small scale lab, but when you want emphasis in a larger scale, it becomes very, very tough. That is one of the major challenge which I felt. So how we can overcome? We can overcome by validating this process. And I put the work on machine learning. So machine learning is especially suits well for when you use it your data management, data mining, then bioinformatics, cheminformatics, those types of data. When you want to use it, first you should know what is this machine learning. Then only you can do this and facilitate analysis of the multi-tissue and multi-organ data. So when you have these data, all published data are available, sit in the computers and try to learn it, where the alterations are taking place, whether I can pick up some of these data into my line and prove it in the laboratory. And integrated pedagogy is very much needed. It is not only my pharmaceutics or pharmacology analysis. I want to have a networking with all the departments. That is very, very important. And those who are interested, instead of curriculum, they can put them in the elective subjects, and those who are interested, they can take up this as their topic of subject of interest. Okay. To conclude my talk, the future aspects of therapy looks more into biomolecules. So that means we have to know about the structural biomolecules, how it functions, how it deliberates, and how it modifies, how it, how it executes its function, and where I can intervene, and how with those interventions with my drug or chemical or my plant material or my miRNA or RNA, where I can do it, or when I do the interventions, what type of biomarkers I can choose it to show the recovery or intervention is proper or appropriate. And as a teacher, as a pharma professions, my humble request is we have to change our teaching a little bit slowly into a newer techniques involving informatics and integration of omics based strategies, which might help our students to get much opportunities, because we are very strong in drugs, we are very strong in analysis, we are very strong in pharmaceutics, this formulation development. As because of these aspects, the drug aspects, we add a little bit more on this flavor to our students will definitely benefit them. So with this, I conclude my talk. I thank the organizers, our Vice Chancellor of DIPSAR, Dr. R.K. Koyal sir, and other team members, Dr. Rajini, for their opportunities given. I hope I reached the audience. Thank you, one and all. Over to the moderator.
Thank you, sir, for such an informative talk. And you have explained everything with very relevant examples. If I say, you have rightly said that omics technology plays a key role in pharmaceutical research. Today, when we look about actually drug discovery, diagnostics, targeted drug development, it is possible with the use of high throughput analytics and approaches and bioinformatics based on omics.
Yeah.
And if we compare India with other countries, even we found that research is going on in India, but it is very fragmented, it is very fragmented. So we need to come together. And your topic is topic was very relevant. The use of omics technology in pharmaceutical sciences is important, and inclusion in curriculum will provide a pathway to precision medicine development and biologics through pharmaceutical professionals. So it was very nice. And now, Dr. Lata, ma'am, it is up to you. Please.
Unmute Casey. Participant, if you have any question, you please type in the chat box. Sir, I want to ask, in your college, sir, relevant to proteomics, whether you are doing any research, sir?
Yeah, actually, we are having a, see, we are linking with biotechnology department of PhD Tech. And so definitely we are doing all except analysis. So we are doing a little bit on these lines, that is protein identifications, characterizations, and even we got one recent projects were linked with PhD Arts and Science on monoclonal antibodies, how it can be helpful in COVID-19 cases. So that we are doing it a little bit on this line. And because we have a team like biotechnology, arts and science, and pharmacy. So we are doing it, ma'am. Okay, if anyone of you are interested, you can always visit us, no problem.
Okay, sir. DIPSAR also we are having biotechnology department. M.Pharm biotechnology is also there. M.Pharm pharmacology is also there.
Yeah, definitely can integrate, madam. With these departments, you can integrate. We have very beautiful work. You can think it of. It's become very quality science can come out, ma'am.
That's why whatever basically whatever is possible that we can start, and then we can think about the research and all. No? That's why basic thing what all the thing you are doing that I want.
Yeah, sir. Good afternoon. This is Dr. Khobragade, Deepak Khobragade. Sir, any facility regarding PRP isolation or studies? Platelet-rich plasma.
PRP, that is for you need about the cells isolations.
Yeah, and lyophilization.
Yeah, because we need fax. And it's a fax machine along with their sorters, and we have a sorter markers. If you want to put those sorter markers, go isolate those cells, and we grow it in a separate way. But we have basic fax, but we don't have sorter.
And sir, what about the identification of key factors of PRP? So for that, you have any facility?
No, we don't have it.
Okay, sir. Thank you.
Yeah, thank you.
Any question on the participant side? Do you want to ask any question?
Okay, if there is no questions, I thank all the organizers, and you can very well reach me through my email, and I'm very much thankful to the organizers. Thank you, ma'am, and thank you, sir.
Thank you, sir, for this great presentation. Thank you.
Thank you, ma'am. Thank you, ma'am. Can I sign off?
Sir, thank you so much for wonderful presentation, and I really appreciated how you integrated the thing that how it is relevant in the curriculum as well as the research, and that we have to learn to teach. I am sure that the audience benefited immensely, and I'm also thankful to you, sir, that you took up the accepted to be part of this program at the first request.
Yeah,
It
thanks a
is
lot.
your total
Because
humility.
because I know sir very well. I visited your university. I was there, so I came twice, thrice. So it's you're part of my. Like I feel it's my home.
Thank you, sir. This is your humility and your kind gesture. If Dr. Sachin is there, we can share the certificate, sir. We have prepared a small certificate as a token
Okay.
to appreciate your. Thank you so much, sir. I'm sure
Thanks, sir.
Dr. Geeta will join us in felicitating Dr. Ramanathan. Madam, please.
Yeah, thank you, sir. Please accept our thanks and accept the certificate from our side.
Sir, I will be in touch with you on mail.
Yeah, please.
And, thank you so much, sir.
Welcome, ma'am. Welcome, ma'am. Welcome, ma'am.
Thank you, sir.
Yeah. Thank you.
Thanks for joining.
Dr. Geeta, I can see that the next speaker is here. So
Yeah, sure. Next, I think next speaker is Dr. Trista Dixit.
Oh, sorry, I'm just interrupting you. It's Reema Sani Mediratha on basics of intellectual property.
Yeah.
Okay, ma'am. So first you can introduce Dr. Reema.
Yeah, sure. So I'm again pleased to introduce second speaker of this session, Miss Reema Sani. And Miss Reema Sani is a registered patent agent with Indian Patent Office. She has done his postgraduate diploma in IPR from National Law University, Bangalore. She has having experience of more than 7 years in formulating IP strategy and advising many SMEs, startups, and individuals. Her key expertise areas include IP management, technology transfer. I welcome you, Ms. Reema Sani, and now over to you. You can start your lecture. And I think this talk is again going to be very useful, intellectual property rights, because most of the Indian researchers, they are very innovative, they are doing their research, but sometimes they go for publications rather than patenting their ideas and innovations. So it is going to be useful for students as well as faculty. Thank you. Please.
First of all, thank you to the organizing committee, DIPSAR, for giving us this opportunity to join this session as a speaker. So I'm just sharing my slides. Please let us know if these are visible to you.
Yes, ma'am.
Yeah, it's this.
Okay.
Yes, ma'am.
Thank you so much. So today I'm presenting Introduction to the Intellectual Property Rights. It's all about basics of IPR and what are the benefits of filing IP rights. So what are IPR? If you think out of the box, generate ideas and own them is called the intellectual property rights. So these are the few pictures of IPR, just like the patenting diabetes drug, trademark, copyright, industrial design, trademark, and geographical indication. So what is the nature of intellectual property? Intellectual property is basically the creation of mind. It is our intellect, these are our ideas which we expressed in an tangible form. So it is intangible property like house property, These are the exclusive rights which are given to the creator or the inventor of the IP on disclosure of their idea to the government. IPR is territorial in nature. Why we say it is territorial in nature? I can brief you about territorial nature of IPR in case of patent. Nature of IPR is basically they are negative rights. Why they are called negative rights? Because by filing IP rights, we prevent others from using your rights. IPR is a time-bound property. So every IP is granted or registered for a limited period of time, which we will discuss one by one. As IP rights are given by government, so they are legally enforceable. For example, if anyone is using your IP or copying your idea and you have already protected your IP, then you can easily protect your IP by taking any legal action against the infringer. So what are the benefits of filing IP for an academic institution? So IPR is the new way for academia to disseminate its knowledge and create solution for the good of society. So I can give you the best example. In the era of COVID-19 outbreak, you must have seen all the academic institutions, they come out with some COVID-19 solution, for example, or the mask, diagnostic kits. Most of the academic institutions, they come out with the COVID-19 solution. So IP is just like they are adding something which is good for society as well. IPR not only helps innovator to protect their invention, but they also provides better collaboration and funding opportunities as well. For example, if your IP is worth, if your invention has some market potential, then you can file for funding opportunities as well. For example, BIG scheme of BIRAC, you can apply for that. IPR is an asset for any academic institution. It helps in building brand value of an academic institution. For example, if you want to get admission in any academic institution, what will you see? You check the ranking of that academic institution, and one of the factor is IP for it contributes to the ranking of any academic institution, because IP portfolio, like how many patent application an academic institution has filed, how many trademarks, copyrights, and industrial design. So IP portfolio, it acts as brand value for any academic institution as well. It promotes innovation, collaboration, and student entrepreneurship, because IP, it promotes students or main inventors to build their startup and start their business venture as well. So from IPR, it is the new career opportunity for faculty, staff, and students. And it is the new sources of revenue as well. Because by filing or protecting your IP, you can start your startup, and you can further develop your technology with the help of the collaborators or the other funding schemes. And also you can translate your IP. You can do technology transfer and earn revenue from your technologies as well. IP and TT, technology transfer, it promotes university academic spin-off as well. Spin-off is basically the business venture which is initiated in any academic institution, which is started by their faculty or students, and they earn profit from spin-off as well. What are the drawbacks of not protecting your IP? If you have not protected your idea, then anyone can copy your idea, and you can't take legal action against the infringer. And in a way, you are creating competition for yourself as well. Why? Because any big company can copy your idea because you have no any evidence that it is your, and because you have no filed any patent application for your idea. So any big company can take your idea, can manufacture the product and start selling in the market. So indirectly, you are creating competition for yourself in the market. Types of intellectual property. There are a few types of IPR like patent, trademark, copyright, industrial design, trade secrets, geographical indications, layout circuits. So what are patents? Patents are exclusive rights which are given to the inventor or the assignee or the patentee by disclosing its invention to the government. So earlier patent was granted only to the process. Now it is available for product as well as for process in India itself. The time period for grant of patent is 20 years from the first filing of patent application. Okay, let's discuss about territorial rights. Patent rights, like other IPR, are territorial rights in nature. For example, if you have filed patent application in India itself, then you can only enjoy all the rights in India itself. But if you want to enjoy the patentee rights in US, then you have to file different patent application in US as well, because patent rights are territorial in nature. If you have filed patent application in India and not filed in US, and somebody is copying your idea in US, then you can't stop them in copying your idea because you have not protected your idea in US itself. So for protecting your idea worldwide, then you have to file patent application in different countries as well. Patent rights are negative rights in nature. Why? Because you are stopping others from copying your idea. You are not giving consent to others for using your idea. Like other properties, patent rights, patent can be sold, licensed, mortgaged, inherited, and transferred. Rights of a patentee. Patentee has exclusive right to manufacture and use the patented technology. Patentee has exclusive right to sell the patented invention in the market, and the patentee can stop others from using, manufacturing, and selling the patented invention. Patentee can prevent others from importing the patented invention in the country in which there is patent protection. So how patents are beneficial and how it is beneficial to file patent applications? Why? What is the purpose of patent? So granting a patent for an inventor, it is a motivating factor. It adds incentive to the inventors. It encourages research and development in the country. It brings the technology to the market. It promotes technology transfer, basically the translation of IP. It promotes commercial exploitation of technology at the fullest. What are the patentability criteria? If you want to file any patent application, there are 4 criteria which you have to evaluate if your technology is worth of having these 4 criteria. The first one is novelty. Novelty means your patent application, like your invention or your idea, should not be in the public domain. It is said that only one paper can kill the novelty of your invention. So it is always recommended to refrain from publishing your idea before filing patent application. Second most important criteria is non-obvious and inventive step. It means that your invention should not be obvious to the person who is skilled in that art. And it should have inventive step, means it should be technically advanced It should have the technical advancement than the already existing technologies in the market or in public domain. It should have industrial application. If there is no any use of your invention, then it is not worth of filing patent application, because it will be objected by the examiner as well. The fourth most important criteria is patentable subject matter. Your invention should lie under the Section 3 and Section 4 of Patent Act 1970. So I have picked few subsections from Section 3, which are important for the pharmaceutical patent application. The subsection A of Section 3, it said that any invention which is frivolous and which is contrary to the well-established law is not patentable. For example, there is nothing like perpetual machine. For running any machine, it needs input. Second, the subsection B of Section 3 said that any invention which is contrary to the public order or morality and which is causing prejudice to the human health is not patentable. For example, any illegal drug which is causing causing serious prejudice to the human is not patentable. Subsection C, it said that discovery of scientific principle, just like any Newton law, Einstein equation, Pythagoras theorem, these all are the scientific principles. So anything which is based upon on these scientific principles are not patentable. Subsection D, which is the most important section in case of pharmaceutical. Any new form of a known substance or new property of a new use of a substance which does not result in the enhanced efficacy of the existing substance, it's not patentable. We will discuss it later on with the interesting example. Subsection B, it is subsection E of Section 3, it said that mere admixture resulting in the addition of property of known substance, it's not patentable. For example, if you mix this proportionate of few salts, then it is not patentable because there is nothing innovative in that. You are just mixing 2 or 3 salt in a proper proportion. Subsection I of Section 3 said that any method of treatment of human being or method of animals or any method to increase the economic value of animals or plant is not patentable. Diagnostic methods are not patentable, but diagnostic kit, these are patentable. And you must have seen the diagnostic kits which are invented during the COVID-19 era. The inventor have filed patent application for them as well. Subsection J, plants and animals in whole or any part thereof is not patentable, but the genetically modified microorganisms, they are patentable. So Section 3D, as I have already mentioned, that the new form of existing substance which does not result into increased efficacy of the existing substance is not patentable. So there is a landmark judgment in the case of Novartis anti-cancer drug Glyvic in India. Novartis International, which is a Switzerland-based company, they filed patent application for Glyvic medicine, which is used to treat chronic myeloid leukemia and gastrointestinal stroma tumors. It invented from the beta crystalline form of imatinib mesitylene. This drug is famously used in the treatment of cancer. And moreover, it was patented in more than 35 countries. So beta crystalline foam was derived from the imatinib mesitylate, means it was the new form of the existing substance. The Novartis was not able to Novartis was failed to prove the therapeutic efficacy of beta crystalline form of imatinib mesitylin in front of Supreme Court. So why I'm quoting this example, because I'm not giving you all the detail of this case. I'm just telling you why these new form of known substance is not patentable. So this is the way to prevent evergreening of patented products by introducing minor changes in the existing substance. So Novartis had filed appeal, and I'm just letting you know how the case was started and what was the history of the case. In 1997, Novartis filed a patent application in India for the drug Glyvac. In 2005, India introduced the Indian Patent Act, preventing the frivolous patent. And before that, only there was the process patent, not the product patent. In January 2006, the patent controller in Chennai denied Novartis a patent. In May 2006, Novartis challenged the Indian government and 4 other companies in the Madras High Court. In August 2007, the Madrid High Court ruled against Novartis' case. June 2009, the IP Intellectual Property Appeal Board rejected a fresh appeal by Novartis. In August 2009, Novartis approached the Supreme Court of India. And how Supreme Court of India concluded this case. These are few excerpts from the judgment of Novartis case. In the case of Novartis, Novartis failed to prove the therapeutic efficacy of beta crystalline form of imatinib mesitylin is more as compared to the therapeutic efficacy of imatinib mesitylin. Means the beta crystalline form was not had not the therapeutic efficacy more than the existing substance. So that the application of Novartis for patent rejected by Supreme Court. The judgment was given by the Honorable Supreme Court is to prevent the evergreening of patented products by introducing minor change. Why? To give relief to those who can't afford the life-saving drugs, as these pharmaceutical companies, they sell such life-saving drugs at a very high price, hence unaffordable for the common man. So this was the main mandate behind the rejection of this Glyvac patent. Now come to the Indian patent filing system. So there are two ways of filing patent application in India itself. First is you can directly file the complete specification with IPO, or if you think your invention is not complete and you can further develop the technology, then you can file provisional patent application with the Indian Patent Office, and you can save the priority date of your invention. After filing patent application, if you have filed provisional patent application, then there is a 12-month timeline in which you have to file complete specification. And after filing complete specification, from the 18 months from the filing of your application will be published by Indian Patent Office. If you want to get your application published early, then you have to file Form 9 for a request for early publication. In case of patent application, it is mandatory to file request for examination. If you will not file request for examination, then your patent application will not be examined by the examiner of Indian Patent Office. So there is a deadline of 48 months from first filing of your patent application, during which you can file a request for examination with the Indian Patent Office. But it is always advisable that you can file request for examination with filing complete specification itself, so that your application will be examined early. In case of startup, they can they have the opportunity for filing expedited request for examination as well. After filing request for examination, the first examination report, it will be issued by Indian Patent Office. And there will be a few objection in that examination report. And there is a timeline of 6 months in which you have to file reply to the response to the examination report. If you will not file response to the examination report, then your application will be abandoned. And you can extend the deadline for another 3 months as well. Okay, and after that, if examiner is convinced with your response, then you can directly grant your application. If is not convinced, then you will be called for the hearing, and in hearing they can refuse your application, or it depends how it is worded. It can be granted by the examiner as well. So there are two prosecution as well. After publication, there is a pre-grant opposition, which anybody can file the pre-grant opposition after the publication. And there is a post-grant opposition as well after the grant of your patent application. Types of patent application filings. So the ordinary application filing, Paris Convention, PCT application. Ordinary patent application is basically the application in which there is no any priority date. You are directly filing patent application, just like the provisional patent application you have filed without claiming any priority date. Paris Convention, it is an PCT application. These are the treaties. Paris Convention, it has the 177 members to this convention, and it is the oldest treaty and the most adopted treaty by all the countries. And PCT, Patent Corporation Treaty, and 153 countries are members to the PCT. So if you want to file patent application in abroad as well, after filing your patent application in India, then there are two ways by which you can file international patent application. One is Paris Convention and second is the PCT filing. What is the difference between both and why PCT application is beneficial than the Paris Convention? Okay. In Paris Convention, if you have filed provisional patent application and after 12 months, there is a deadline of complete specification, then you have to file Paris Convention, through which you can file your application in abroad in the countries which are member to the Paris Convention. So deadline which is given under the Paris Convention is 12 months from the first filing. But in case of the PCT application, you have PCT. First of all, I would like to clear that PCT is the not granting authority. It is the route from which we are filing our patent application in abroad itself. So under PCT, you can file PCT application in 12 months. And then there is a deadline of 30 to 31 months, depend upon the country, from the first filing, in which you can file, you can go to the national phase. So in PCT route, you can file your patent application in one language. There is no need to file separate documents. And the search report, which is issued by PCT, is very beneficial for the assignee or the applicant of the patent application to decide if its invention, if his or her invention is worth of filing patent application in other countries or not. So if you want to file patent application in more than 4 and 5 countries, and you are not sure that at the time of filing that you can file in other countries, then it is always recommended to file through PCT route. Okay, now discuss about few interesting example. There is one example, which is a word non example, which is the academia which become billionaire by filing patent application. And it shows the significance of filing or protecting your IP. So you must have heard about the beverage named Gatorade. So researchers at the University of Florida, they began to study why the school football players were being affected by the heat. And they invented a new drink for the athletes, and it was Gatorade. And then they sold it to the beverage company, which is Stockley Van Camp in 1967. And company agreed to pay the team $25,000 in addition to a small bonus and a 5-cent royalty as well on every gallon sold. And because the drink was invented by the student of University of Florida. So according to the IP policy of that university, 20% of the royalty, it belonged to the University of Florida. You won't believe, as per ESPN report in October 2015, royalty surpassed $1 billion. So this is the most important example which I could have quoted, how the university become billionaire by filing patent application and further transferring the technology to a company. So there is another case study of inventors who did not get rich. I think you all are aware of karaoke machine. It is nothing, it is empty orchestra. I have seen so many people who have discovered their new talent of singing in the COVID-19 lockdown. So anybody knows who invented karaoke machine? It was a Japanese businessman, Daisuke Ine, who used to make money by playing drum in a bar. And one time when he could not play the band, then he put the backing music on the tape. It was empty orchestra and it worked out. And later on, he made 11 karaoke machine and which he leased out. But he did not file any patent application for this invention. If he would have filed patent application for karaoke machine, then he could have transferred the technology or earned revenue from that. So this is the case in which inventor, he could not become the billionaire by filing patent application. So this is very interesting thing. Innovation Technology Transfers Office initiative, which is Humse Poocho. This is a web-based one-on-one session to answer queries which are related to IPR and technology transfer. And this is open to the faculty members, students, startups, and entrepreneurs. So every Friday, we are giving opportunity to the faculty members, student, and startups, and any entrepreneur to book your slot with us. And our experts in IP and technology transfer, they can answer your queries. So please go to our LinkedIn profile, and if possible, I will share this link and book your slot for if you have any doubt regarding your technology, like how to make the IP formulation of IP strategy, any kind of advisory or any doubt. What are trademarks? Okay, this is very good statement in which, So John Stewart is chairman of Quaker. He once said, if this business were split up, I would give you the land and brick and mortar, and I would take the brands and trademarks, and I would fare better than you. So statement by John Stewart, it tells us about how significant is to file trademark application for your business venture, because it helps in creating brand value for your business. What are trademarks? Any sign then that individualize the goods of a given enterprise and distinguish them from the goods of its competitor is trademark. For example, everybody is familiar with the brand name Apple. When you go to buy any phone, then there is Apple phone, Samsung, and Nokia, or any other Android phone. You will pick. Why you will pick Apple? Because you know the quality which Apple brand is promising to you. So basically, Apple is a sign or the trademark which individualize the goods of a given enterprise, and it distinguish the product from the competitor product as well. So it is the identity of any businessman in the market. It helps in building brand value. And the term of trademark, like patent, it is 20 years. In case of trademark, it is 10 years, but you can file renewal for trademark application as well. So it is always recommended to protect your trademark before launching your business. So I'm giving you a very interesting example of McDonald's and Starbucks. McDonald's and Starbucks, when they decided to launch their business in India itself, first of all, they protected their IP portfolio in India first. And after filing IP protection in India, they started their business in India. So this tells us about how filing trademark is important in building brand value. What can be trademark? Like patent application, trademark should also be unique. It should not be in public domain, and it should not be obvious, or it should not relate to the quantity, quality, or anything of the product. It should not hurt the religious susceptibility of people, and it should not be prohibited under the provision of improper use of words. For which we can file trademark application. Trademark can be filed for any word mark. For example, the Gmail, Coca-Cola, these are the words. It can be filed for the logos like Apple, BMW. It can be a combination mark, means the logo plus word mark. It can be a colored mark, and any packaging mark, product shape, or rarest examples are sound spell marks are also available. Now come to the next IPR, which is copyright. And copyright, it is the bundle of exclusive rights which are given to the creator of original work. So means it should not be the copied work. It is given, the copyright are the exclusive right which are only given to the creator of that original. Copyright protection does not depend upon the registration. Like if you have written any book, if you have written any PhD thesis, then copyrights belongs to you as well. So it is not mandatory to file copyright application for your work. The copyright law protects ideas expressed in a tangible form. For example, in my mind, there is a poem, but I can't protect it through copyrights until or unless I write down that poem on a paper. So it is not given for the idea, but if you express your idea in tangible form, then you can protect it through copyright law. Okay, when author of the work is an employee and the work is made in the course of the employment, then the author is not the owner of copyright. Why? Because the employer has paid for writing that work to the employee. So the rights will be with company itself. But yes, its name will be mentioned as an author on the book. So as I mentioned earlier, copyright not on idea, but on expression of idea. So these are the examples, the novel of Chetan Bhagat and how the movies were made on Three Idiots, on 5 Point Someone, means the director has expressed the idea of Chetan Bhagat in a different form. But yes, director had given due credits to the author as well, but there were some disputes as well after the release of the movie. Okay, and same the Shakespeare novel Hamlet. Heather was the adaptation of this novel as well. So this is the best example of how you can protect copyright because novels are in public domain, but you can express that idea in different form, but by giving due credits to the creator of or the owner of copyright, or you can take permission or consent from the copyright owner. Copyrightable matter, any literary work, poem, essay, everything they comes, or any book, these are the copyrightable matter. Any musical work, artistic work, sound recording, these all are come under copyrightable matter. Rights under copyright law, the copyright owner has right to reproduce his work. Copyright owner has right to distribute the work, and he can adapt or convert his work in any form. Right to make derivative work, and the copyright owner has the exclusive right to public display of his work. Now come to the next IPR, which is industrial design. It is very interesting IPR because it is like how industrial designs are adding value to any article, and how important is that. I can, if you are going to buy any chair or any sofa set, there were two chairs, chair A and chair B. Both are very comfortable. Both are available on same prices. But you will buy chair B. Why? Because the design of chair B is very unique. If you have to give more money for that chair, you can go for that as well. This tells us about how industrial design, it adds value to that article. Product design input, it contributes 60% to the wealth generation. It is the value addition, as well as influences 50% of the inbuilt quality of its value chain. And thereby it contributes significantly to the competitiveness of the product. It means the industrial design, it adds value to that existing product. So industrial design, it protect design that are applied to the articles. Not if you have do something, some design on a paper that is not eligible for filing it an industrial design application until or unless you have applied that design on the article itself. Means you have do that design on a paper and then you applied that to the chair if you have prepared for the chair. So industrial design, it is one of the category of IPR where the design system, it focus on the aesthetic feature of that article, which add visual value, means it is very appealing to your eyes. The ornamental or aesthetic aspect of an article that enhances the visual appeal of that article. It can be filed for any shape, configuration, surface pattern, the color or line, or a combination of line which are colors or which are applied to an article. The design can be of 2-dimensional or 3-dimensional. And like other IPR, the timeline for protection of industrial design is 10 years from the date of registration, which can further be renewed. So industrial design can be applied to any device, or it can be applied to any component of the existing device as well. So these are some interesting examples of industrial design. For example, the Coca-Cola bottle. And the most interesting is the iPod design patent, iPod design. because earlier people used to keep songs in pen drive. Okay, then Apple, they launched iPod, in which the function of iPod is similar to the pen drive. It is used to keep songs, but because of its design, the cost of iPod is 3 or 4 times higher than pen drive. But because of the aesthetic sense, the aesthetic feature of that iPod, it is very appealing to the eyes, and most of the people, they bought iPod. So it tells us about the significance of industrial design and how it adds value to the article. Okay, publish to perish, patent to prosper. So our Honorable Prime Minister has said, my motto for the young scientists in this country is to innovate, patent, produce, and prosper. These are the 4 steps which will lead the country towards faster development. So IPR detect as knowledge economy and which contributes toward GDP of India. So it depends upon you to publish your invention and kill. By publishing your idea or invention, you can kill your invention, or you can maintain the confidentiality of your idea and file patent application for your idea. And after that, you can convert or translate your invention by transferring the technology, or maybe the other company to whom you have transferred your technology can further develop your technology and sell in the market, and from which you can generate revenue in the form of royalty. So this tells us about we should always refrain from publishing your idea before filing patent application. Significance of patent searches. So it is always recommended when you are ready with your invention and interested to file patent application. First of all, you should evaluate your technology. You can contact any IP professional. Why I'm saying that? Because from the Google patent few hit, you can't decide if your patent application is novel or not. Because IP professional, they have the paid databases. They can go for the extensive research for your technology. So you can go for novelty search and patentability search. They can tell you about if your invention qualify all the 4 criteria of novelty, inventive step, patentable subject matter, and industrial application. Why I'm stressing on going for novelty or patentability search before filing patent application? To avoid reinventing of wheel. Because if your idea or invention is already in the public domain, then it is not worth to file patent application. Because if you will file patent application, then it will be objected by the patent office. So it is always advisable, first go for the patentability search, and there is the add-on advantage that you will come to know who all have filed the patent application in the similar domain, what is in the existing market, and what are the gaps left in which you can do your research and fill those gaps. And the second most important is to avoid unnecessary expenses, time on filing patent application. Because filing IP patent application is very expensive. So it is always advisable to go for novelty or patentability search before filing patent application. Formulation of IP strategy, where to file patent application. So you can take the advice of expert in formulation of your IP strategy. First of all, if the origin of invention is India itself, then first file your patent application in India and then decide in which country you can file your patent application. If you have filed your provisional patent application, then there is a 12-month of deadline in which you can do market survey as well. First of all, you can, before filing in Indian patent application, you can evaluate your application for patentability criteria. And after that, you can file patent application in India. After filing patent application, there is a 12-month deadline in which you can evaluate the market potential of your patent application, if it is worth of filing in abroad or not. Why I'm saying this? Because IP portfolio is very effective in commercialization of your technology as well. If somebody is, if you want to transfer your technology, then every licensee, they will check how many patent application or trademark or copyright application you have filed for your invention. I can give you a very interesting example for formulation of IP strategy. An inventor has invented a medical device for treating cervical cancer. He filed patent application in India itself. And after that, he realized that there is a market potential of his invention in US itself. Why? On which parameter he decided that? Because the patient of cervical cancer, the rate of cervical cancer is higher in US. And there is no good existing product there. They are not at all effective. And inventor's medical device has a technical advancement, and it has no after surgery drawbacks, and it is available in cheap price as well. From that market potential, he realized that he should file patent application in US itself. He filed patent application in US. He launched his product in India and US in both countries, and now he is earning revenue from technology transfer. So this was all about basics of IPR. Thank you everyone for your patience. If you have any query, you can always write to us at queries.ittu@outlook.com. Over to you, moderator. Thank you so much.
Thank you so much for this very informative talk. Thank you. I think we will take the question at the end of both the sessions, because both the sessions are very related to each other: intellectual property rights and technology transfer.
Sure. Thank you so much.
Thank you. Now, I am pleased to introduce our third speaker of the session, Dr. Dixit. Dr. Tripta Dixit is Deputy Project Manager, Innovation Technology Transfer Office. She is candidate registered technology transfer professional. Dr. Dixit is having more than 5 years of experience in intellectual property, technology transfer, and policy framework at ICMR. She is awarded Howard Bremer Scholarship by Association of University Technology Managers, USA. So I welcome you, Dr. Dixit, on this platform. She is going to deliver a talk on nuances of technology transfer. Now over to you, Dr. Dixit, please.
Thank you so much, ma'am. Thank you for a wonderful introduction. And I thank DIPSAR team and all the dignitaries around for giving us this opportunity to interact with all the pharma faculties that, we would have never, I think, got the chance to interact with. So before I begin with, I have a request that, can I keep my video turned off to have a better bandwidth? And in case, if I miss out for a second or two, so kindly apologize to me. I mean, I seek your forgiveness for that, for the inconvenience, because it's all about internet connectivity. Yeah, so a very warm welcome to all. And it's wonderful to be here with you all and share something that I am really passionate about. For today's session, we will not be talking about pharma or any related subject. Rather, we'll try and understand how we can be benefited or contribute to the society with what we have or what we are capable of developing in future. So yes, we'll be talking about commercialization, technology transfer, and ways to introduce our years of efforts into market. I understand all of us, all the pharma faculties, and there's one thing which, before I begin the technology transfer session today, I would like to share with you all. You will not believe the most successful licensees and the most successful and the most success stories that we have come across so far are from the pharma field, because they have the potential to develop wonderful drug candidates which eventually result into some great medicine, some product which actually support the society. And it is always
Audience, please unmute. Mute your mic, please.
Hello, am I audible now?
Yes, ma'am. Yes, ma'am.
Was I audible since beginning?
Yes, ma'am.
All right, fair enough. Yeah, so I was saying that the most, beneficial patents or beneficial licenses that we have come across are from the pharma background, because they have most potential to generate maximum revenue that a university or a person or academia can think of. So today, let's try and understand, for the timing, I request all of you to forget that what we do. Let's understand how we can commercialize the generalized path to actually transfer a technology. And then once we are done, I would request all of you to get back to your inventories and see what all you have, and apply this particular session to your study, and then see how better we can commercialize. So with this, let's begin today's session. So I would like to share a few calls with you that will give a generalized idea that what people think about tech transfer. So we often get calls and people say, we have received a company's interest in our technology and I want to transfer. So can you please share some agreement or document? So really, is technology transfer signing a document and we are through with it? Well, we'll actually understand that. Another caller says, I have developed a product and we are likely to receive a company's interest. So can you tell all the regulatory requirements we need to fulfill before we transfer? Now, my question here is, are regulatory requirements for technology transfer, or are those confirmatory study or the standard guidelines which have been proposed by Government of India, to actually see the suitability of your research on some specified criteria to be used by humans and environment? So we really have to be clear on that front. Then another caller says that, I have a very enthusiastic faculty say, I have developed a wonderful product, let's say a drug candidate, and I know there's a company who really wants to take the technology. So in all these three calls, we have one thing in common. One is the technology. So here, when I say technology, you all can actually think of a new formulation or a candidate, that if that helps. So we have a common technology. We have a technology and the intent of transfer. But is this sufficient for technology transfer? So with this thought, let's understand nuances of tech transfer. So for today's session, we will be covering what, why, and significance of technology transfer, how and other aspects, that means different modes and tools used in tech transfer, some common strategies. And for today's session, I have just taken very recent case studies that ITTO Innovation Tech Transfer Office has worked upon. So ours is a very new establishment, and we have actually had a few success stories in this COVID time. So I'll just give you an example from a faculty perspective that, how you can think about tech transfer and essence of technology transfer office, because all of this is happening at technology transfer office. So we should also understand what is it that they do, what is it that they're capable of, and where all they can support you. Then we are going to, before I jump into this, there are a few terminologies that I would request and I really hope that most of you are aware of it. These terminologies are being, frequently used and repeatedly used in the innovation ecosystem. So innovation, again, a new concept, a new idea, a new discovery. But then or an improvement of an existing idea. Invention has been taken up in detail by Rima, my colleague. So invention says that you have a product, you have a new idea, but then it should fulfill all three patentability criteria. Means it should be novel, it should be inventive, it should have applicability. Then translation is the process of conversion of idea or any new drug candidate into some good or service, or here we can say a product which can add value to the customer eventually. Entrepreneurship, an activity of setting up your own business. And then technology transfer, I call it a delivery channel with right skill set and tools for smooth translation of science into a commercializable product. So with this, let's understand what actually I'm talking about. So it's a translation path. It is very important for all of you to understand what a translation path is before we actually looked at, where all technology transfer plays a role and how. So let's understand a generalized translation path. I'm sure all of you are aware of it. So I have tried dividing the translation path in general in 3 different stages: developmental, manufacturing, and marketing. Because here all we are focusing upon is commercialization, how to enter the market. So let's see, at developmental stage, if we talk about, let's say you have conceived an idea, that means you have actually conceived that there's a drug discovery and we found a lead compound. So now what next? So we would like to convert it first into something which is tangible and on which we can actually perform the study. So we will think of having a drug development, or think of having a prototype. We have, let's say, developed a prototype. Now, can we actually, is it sufficient to have that prototype? Just to confirm what we are actually claiming, we need to have validation studies performed for that. So at developmental stage, this is wherein we prepare prototype, we perform different validation studies to be sure of what we are committing, that is what we are claiming is right. So this is a stage when we are most likely to identify intellectual property associated with it. Intellectual property can be in any form. It could be a trademark. You want to give a brand name. You want to give a name to the particular lead molecule. So it could be a trademark. Anyway, then it could be a patent. Again, you have identified some molecule. You have utilized those different bioinformatic tools, and then you understand that it has some novelty in it. It is being identified for the first time. So we would like to have a patent secured, or a new process of. So we can also have a process patent here. So all the possible intellectual property, I would also say, in not just in pharma, but in other streams, we also have copyright available. We also have design of a device that we have invented. So this is the stage when we are most likely to identify that, and it is always advisable to protect it. Once we have protected intellectual property at this stage, we have added strength and value to our technology. Now we are free. We are free of the fear that nobody is going to infringe and nobody is going to copy it. It cannot be infringed, right? So we are more confident to move ahead in the process. Now, if you move further a little up, if you look at technology transfer, we all understand technology transfer only happens when we have something tangible with us and we would like to give it to company for upscaling and manufacturing. But then why we are covering the technology transfer at this stage? Let's say you have identified the lead molecule and now you want to perform some bioinformatics study for that, for which you do not have the lab facilities, neither you have the competency. So you would like to seek those competency from some bioinformatic expert, and here we call that consultancy. So you would like to seek the consultancy, and we can execute a consultancy agreement at this stage. So that's another type of tech transfer. Technology transfer definition will be taking in the upcoming slides after 2 or 3 slides, and that will actually give you a clear idea that here even exchange of skill set is also a transfer. So here we are seeking his skill set, his expertise for developing that particular product. So that means that's another type of tech transfer. Then let's say, like I said, that you don't have the facility as well. So apart from consultancy, we can collaborate with some other organization which can help us validate it. So that means we are collaborating, we are co-developing it. So we can again have a co-development agreement here that we are together developing it. You do some part, I'll do some part. So that way. Now let's look up. It says regulatory compliance also fall into this. So it is always said that, regulatory compliance start from day one, right from the day you design your study. Because there are certain standard regulatory documents and certain standard guidelines being issued by the government and being issued by different regulatory authorities, depending upon the kind of product and the domain, that, it is always advisable to look at them when you start your research. Why? Because you might not understand the significance now, but when you take the product and you try and hit the market, you say, you go to a company and you say, I have developed this and let's enter the market. Let's take the regulatory approval. And when you apply for regulatory approval, the regulator will say, you never looked into those SOPs, those guidelines that we have issued right in the beginning. You should have looked into it, that what kind of composition should have been there, what should have been the different steps involved in it. So now you're supposed to repeat the entire validation study all over again. So what is it going to result into? Delay in the process of commercialization, wastage of resources, wastage of a lot of efforts that we have invested. So just to avoid all of that, just to be very confident when we are moving ahead in the translation path, we should actually look into it every stage. Now moving on to the manufacturing, second stage. Why do usually we look out for a manufacturer? Because we don't have the upscaling facility, or we don't have more competency in in manufacturing it. Like we require more manpower, we require a proper setup, a manufacturing site for doing it, and we don't have that. So let's say till some small batches study we can perform at our end in our organization, but again, for further studies, for upgraded studies, for more upscale studies, we require a bigger facility. So we look out for a manufacturer. Now here, what kind of tech transfer comes into picture? That, you know, when, let's say, there are times when you want to actually apply, you are actually working on a candidate molecule, and then you understand that it might require clinical trials later. So you understand right from day one that, you know, there's going to be a lot of investment involved in it, and you don't have even their investment to put in. So better to license it to a manufacturer who can actually you know, take the responsibility of taking the clinical trials ahead, and who can actually seek all the regulatory approval, can introduce into the market, and can also manufacture the batches. In case somehow your product might not fall into that category, that, you know, it requires a very expensive regulatory approval or clinical trials later on. So what you can simply do, you can give away some part to the manufacturer, and you can say that let's execute a contract manufacturing license. You contract some pieces for us, we can test it at our end, and then we'll see whether we have to license it to you or no. So here the other path which faculties at times they take is spin-off. They also you know, believe in case the technology, the molecule has multiple applications, so they might even have a spin-off and might not give the right to manufacture, or they might not license it to the manufacturer. They might say you only contract manufacture and we will sell it. So there are different ways of going by it. Then again, for the manufacturing, to set up a site and you know, to perform other quality studies, so you also require regulatory compliance for that, which a manufacturer is supposed to obtain. Moving further into the marketing stage, when we enter the marketing side, so we marketing stage, so what we actually look forward to, we look forward to its penetration in the market, that our product gets absorbed in the market, and we also look at times that, you know, how better we can expand the business. In case you are a spin-off, you're a startup and you have done it of your own, so you would like to actually expand your business. Or in case it's a company and, you know, they want to actually expand the business and they have taken the technology from you, so they will actually give it away to the different distribution channel that you go and sell in the market. So expansion and penetration are two things which actually happen at the marketing stage. Now let's focus on these black bars, the black thick lines. At development stage, it is thick, it is the thickest. At manufacturing, it is a little thicker, and in marketing is thick. So when I say at developmental stage, so at this stage, the involvement of the faculty or the inventor is the max, because they are the one who are devising the SOPs and the different dossiers they are preparing to be given to the company, and they are actually validating it. They are preparing the prototype. They are actually seeing that, you know, what could be different validation studies or optimization studies required for a particular product, and what will be the scale up requirements. So they are jotting down everything. So this is a stage where they are, you know, mostly they are involved. And they put in a lot of their efforts. Once they transfer it, so there are times when the company, they are already into the same business, into developing the same you know, product line, so they might not require your consultancy. But many a times the companies require great hand holding from the inventor, from the academia, so they might come back to you and say, let's execute a consultancy agreement, please be on board with us and help us through the entire process, that, you know, QC check and everything, so that we do not miss out on any step, because we all understand it's a very risky procedure to develop a particular drug candidate, and then again it involves a lot of investment. Then in the marketing stage, it is the faculty, they have their least involvement. By max, they can actually be a part of their launch program, or they can just help them market a bit, but then that's the sole responsibility of the company. They do not interfere much. So this was just a highlight of you know, how the translation path works. So now let's understand with this. All right, for today, I've just taken 3 different cases to give you an idea that what different kind of tech transfer we can have. again, I want you all to think not from the farmer perspective, in general, that what different kind of tech transfer we can have. So the first case study, it talks about a spin-off, how a successful spin-off was formed from a university. So this is a example that, you know, even you at your institute, your respective institutes can think of. You should only you should just get back to your institute, look into the policy, and can think of having a spin-off. Let's see how this was done. So it's Kavach Mask again. One of the IIT Delhi textile professor, along with the alumni and some undergraduate student, they formed a team. They identified there was a need. Now he's a textile professor. He identified there was a need and where you know, best he can contribute. So with his skill set, the team created an innovative mask which is at par with N95 masks at almost 1/3 of its price, and the mask is like comfortable for long wearing and reusable, washable. So it was actually you know, reducing the cost that is borne by the customers. So they got IP protected, they got a patent, trademark, and now again, they also require some validation study, but they need not go to CDSCO because for a mask, they do not give any sort of regulatory approval. So they went to CITRA, which is South Indian Textile Research Association, and ASTM, American Society of Testing Material. So whatever relevant from their understanding, whatever they could think of the kind of relevant regulatory or QC approval they can take, they have taken that. So there was a need. It was identified by the professor. He thought that, you know, actually we can contribute a lot into it. They developed a product. There was a technology market fit. Market needed it. The product was good, cost effective. It added value to the customer. It was reusable, cost effective, good quality. So the product was launched under the startup brand. Now, when I say a spin-off of startup, so what matters the most for them is to have their own brand in the market because they want to survive in the market. It's not just one product that they will be launching. Eventually, because they have expertise in textile, they will come up with active textile. They'll come up with different products with inactive textile. So different kind of textile product they'll be coming up. All they wanted to do was form a brand name into the market, and they did it. Now, in such scenario, even your institute, your organization support. So the kind of publicity that was given to them was also from the IIT Delhi. So that way they actually captured a good market. So this is just an example of how spin-off they work actually. Then another example is, again, Corosur is a RT-PCR based diagnostic kit for COVID-19 developed by one of the IIT Delhi professor and his PhD team in the laboratory. Now, the first example says the professor had the mindset of taking up an entrepreneurial journey. He wanted to explore. In this case, the professor said, I don't want to get into entrepreneurship. I don't have time. I would rather focus on my other research products. So I would like to give it to somebody who can actually bring it into the market. So he gave it to Tech Transfer Office that you see how we can bring it to the market. So that's just his perspective that I'm sharing with you. So again, they identified the need in the market with the skill set which they had. They developed a probe-free one-step PCR detection kit for COVID, wherein RNA is extracted from patient nasal throat swab, added onto a PCR plate which contained master mix with required components. Now this PCR plate is again subjected to reverse transcription amplification, and they use a cyber green dye. So it is a probeless, and the detection is based on cyber green. So this reduces the cost. Again, we suggested them get it IP protected. They got the patent. They got ICMR approval, which again adds value to it, that it is validated. ICMR has also proved it that it has good sensitivity, it gives result in less than 90 minutes. It is easily scalable, and it was transferred to 9 companies. Imagine how much revenue that professor would have generated after transferring it to 9 companies. So this was an example of when a professor don't want to take it forward. Now third, another example says that it's basically a LISA-based anti-Crimin Congo hemorrhagic fever. So these were two very highly demanding, the first 2 case studies wherein we really needed those products and the market needed them. The third case studies say that, it's a regional disease, and Crimin Congo hemorrhagic fever usually, means the states of Karnataka, Rajasthan, Maharashtra, they have been affected in past, and the government of India, ICMR, NIV department thought, institute thought, National Institute of Virology, that, let's have something to avoid in case we are again hit by a pandemic in future. or any epidemic in future. So since ICMR has a biosafety level 4 laboratory, this is what I'm trying to emphasize here. Since you have some facilities which the other institutes might not have, since they had the facility, so they thought that let's develop a kit which can be used by any research laboratory at BSL lab 2, because most of the testing centers, they have BSL2 laboratory with them. So again, it was already published, so no IP was filed, but despite that, it was being transferred to a multinational company, and they have again, generated wonderful revenue. So what I'm trying to emphasize upon that, you actually have to see that what is the need, what are your capabilities, and how better we can actually give away from our inventory at this point of time, and let's generate revenue from that. And I think most of the things, they can be commercialized if they are actually developed from a marketing perspective. Then I'll be taking up how. So with this thought, with this background, let's understand now what is technology transfer. It is the transfer of technical information, know-how, performance skills. As I said, know-how, performance skills, technical material or equipment could be done jointly or as an individual to achieve the goal of commercialization into a product or service, to reach marketplace for benefit of the society and for your, monetary benefits. Now, it's not just an activity, but a multifaceted process with multiple stakeholders. I would like all of you to kindly emphasize, understand this multifaceted process with multiple stakeholders. How? Let's understand through this diagram. Let's say you at your university have, you're the faculty, you have developed a wonderful lead molecule, and you walk up, can you actually take it up and actually enter the market like that? Can you actually look up, think of even commercializing into the market? No. Why? Because it's not ready. So we actually have to see that, what efforts can we put in to actually be market ready. So let's move on to the right hand side, which says technology validation. So we will have to actually see that how strong a technology is to enter the market. So for that, we will actually, we will first prepare a prototype, we'll perform different studies for it. We'll see whether whatever we are claiming that is being matched by the validation studies is reproducible. Are we getting the same feasibility study, stability? Are we getting all of that? We'll test at different parameters. We'll test its efficacy, everything, whatever is related to our particular study. And then we'll be sure, okay, our product is good. Then let's explore the IP. Have we actually protected sufficient IP for our technology? Or the moment we'll show it to somebody, somebody can copy it and can actually, make a better product, or can even launch the similar product, and we will not get anything for years of research. So do you want that to happen? So just to avoid that, we'll also try and protect the IP. Once we are sure we have something very strong with us, now we would like to hit the market. But wait, before we hit the market, let's see what actually market wants. Let's move on to the left hand side. It says market validation. Usually, in case of entrepreneurs, when they actually invest into the market, so they first understand the market, right? Then only, so that's when we say that, identify the need, identify the problem, and then give a solution. But usually when we are working in research laboratories, or we are working on something very, very, unique, and we are hoping that we might come up with something unique. So that's when we actually don't have, market applicability to it at first. But now when we have actually identified some things, let's understand that, what is its market relevance? Do we have similar products? Do we have some other competitive products, or there are other companies who are already into this domain? In case there is something of this sort, then let's see that, at how our product is better than their product. In case, our product is not better than their product, then please take a step back, try and improve it, and try and address the still existing pain points of the market. Because ultimately, let's say you have also developed something, you have put in a lot of IP and everything, right? You have put in a lot of efforts. You also find a company who is ready to commercialize. But what's the use if you are not going to get a regular revenue on that? If it is not going to stay for long in the market, if it gets obsolete the moment it enters the market, or if it is not being used by the people, or bought by the people, or absorbed by the market. So there's no point. So just to avoid all of that, it is always wise we can take a step back right now before we introduce in the market, can improve our product, and then enter the market. Let's say now we are sure there's a market which is ready for our technology. We have good technology. Let's hit the market. So can we actually take it and enter the market? We actually have to now identify and understand how to enter the market. So usually in engineering colleges, what is happening these days that, students, they are actually moving towards different IT interfaces, or AI interfaces, sorry, and then they are also trying to come up with some innovative solutions for healthcare. So they try and work upon some devices, or they come up with some platform technology which can have multiple applications. So in that case, the faculty thinks that, okay, I have a good team, I have students who are working with me and they are very enthusiastic, very energetic, and I also have an entrepreneurial outlook, I have a business understanding. Let me take the path of having a startup and spin-off. So in that scenario, faculty thinks of startup spin-off. He takes the path of startup spin-off and enters the market. In other scenario, when the faculty says, I don't have time to enter the market, I don't want to take any of this, I would rather want to put in my efforts in next study, in further research, So he'll say, I give it to the university IP and tech transfer team, and I would tell them, find a person for me, find a company for me who can take the technology, and then I'm ready to give the technology. So we call that license. So he'll give the license to the company. And in return of license, he'll get some consideration, some monetary benefit. Now, if the faculty say, I want to give it to I want to get associated with a good brand, so we'll give it to a large company. We'll think from his perspective. In case he say, I want to give it to somebody who dedicatedly and only work on my technology, so we'll give it to a startup, because we know that startups will dedicatedly work on that particular technology. If he says that no, I want to give it to somebody who already has a manufacturing unit set up for that, so let's give it to an MSME or a SME who will be quickly able to make it and introduce into the market, depending upon what the faculty intent is for the technology. We transfer the technology. That's how we enter the market. So it's just a broader an overview of how technology transfer actually works. Why technology transfer? To research and develop new solution, to commercial exploit, to get monetary benefits, and most importantly, to overcome translational challenges. What are those challenges? We have understood that what is happening at developmental stage, what is happening at manufacturing stage, and what is actually happening at marketing stage. So let's understand that what academia has at developmental stage, at all these stages, actually what academia can contribute to. So let's say a faculty at a pharma institute can have a wonderful scientific knowledge of formulations, understand all the different compositions of drugs, and also good at ideation. Right. And they also have wonderful initial lab R&D facilities. They are good at formulating studies for testing preclinical studies, but they don't know what next, right? So once they had developed this with all this, they want to just give it to somebody, and they don't know how to do that. So there is this valley of death which they face. Moving on to the right hand side, which says industry strength. Industry has wonderful product R&D. They know how to scale up. They have all the facilities with them. They have already had experience of taking different legal and regulatory compliances. They understand the product commercialization strategies. They have different ways to market outreach, but they don't have the initial, the new lead compound which you have identified and with the wonderful potential. So they will come. They would want to connect with you. Again, they have this valley of death. They don't know that which professor to approach, who has developed this. So how we are going to overcome this valley of death? Let's understand that. Okay, we'll understand that, in the next slide. Usually that valley of death is covered by, let's say, a platform wherein you can actually advertise your technology. You can actually give it to people that, kindly look out for a partner for me who can, with whom I can collaborate. So all this work is usually done by technology transfer office, wherein they try and scout for the right industry for you. Again, industry comes to them and say that we want a faculty who has developed this. So once we know that these people had developed these technologies, so we try and connect both of them together. Now, so moving on to the significance of the technology transfer. What is the significance and why should we actually perform technology transfer? So growth of an individual, in case you are the one who has developed the lead candidate, lead molecule, and you have given it to the company, and so you are getting the benefits of the revenue in form of upfront payment and in form of royalty. Now again, the molecule gets converted into a product and reaches the society. So again, the society gets benefited. And finally, we are all buying it and contributing to the country's GDP. So again, the country gets benefit. So everyone is growing. Self-reliant initially, what was being imported, now we are able to develop it here. We are preparing it indigenously. We have better solution, and So we are more resourceful, improved quality of life. Medicine, which was earlier not being afforded by a mass of people, now even they are able to buy it because it is being produced here indigenously, it is being developed here, so it is definitely affordable, less expensive, it is cost effective. So again, they have access to improved quality of life. And supporting new research discoveries, yes, we come up with new research discovery and creating job at every stage of tech transfer. We support in creating new jobs. Whenever we are giving it to a manufacturing company, in case that's a new kind of product, they will have to hire a new team. In case we are having a spin-off, a startup being formed, we are going to have a new team with us. So in that way, we are contributing to job creation. Now moving on to the next slide, which says Essence of Technology Transfer Office. So maybe after looking at this picture, it gives you an idea that, the bridging gap, this is the, it's just an administrative capacity that a tech transfer works into. But no, here I would like to emphasize that we not only work into the administrative capacity, but we first, let's say first we have received a request from academia and he says that I have this particular product. So we will put an effort to understand what is the product, what actually is the requirement of the product, and accordingly we will identify industry partner with whom we can actually connect the academy. So here I would say that we actually work in leadership capacity. We have networking with different translation unit, funding entities, regulatory bodies. We have access to them and we understand their requirements. So accordingly we can guide you, we can connect you with the relevant person there. Apart from this, we support in early stage domain analysis. We perform IP management, tech scouting, business development, investor and resource connect, co-development, other partnership, commercialization, and also other commercialization strategy defining, and also other tools we provide for the commercialization. Now let's, university, you have a research, you come up with some wonderful product and you give it to a TTO. So what is TTO? Look into the technology, see that, at what stage the technology is, how strong the technology is, and accordingly look out for a partner who can take it up, depending upon the investment still required in the technology in the form of regulatory approvals. So after understanding technology, we actually look out for the partner who can actually take it up. So, and then once we've identified the partner, we strategize that, okay, the partner seems to be a startup, and startup cannot pay a huge amount upfront. So we might have to, put it into different milestone payments, or that, once you complete this milestone, then you pay us this much once you complete this milestone. So we accordingly have to strategize it. What strategy we should adopt. So for that, we will talk with the inventor that what do you want from your invention. We'll understand what company can offer for the invention, and accordingly we'll strike the deal, we'll negotiate the deal. Now we'll give it to existing company who can actually put efforts in the product field trial or pre-launch or product launch. We can also have a spin-out form. So these are different ways and how TTO works. Now, different modes and tools in the technology transfer. How do we do that? So far, we have understood what is technology transfer, what are its significance. Let's understand how do we do it. Idea was conceived, we got a lead molecule, we performed different studies, we found that it has a lot of therapeutic activity. We tried and understanding. We did a drug development and we got a prototype with us. We conducted different trials, commercialized a company, and then it was into the market. But how? What are different ways of commercializing? So it could be a license, it could be assignment, could be startup formation, could be co-development, could be joint venture. I'll be taking each one of this individually in upcoming slides. So here we'll understand different tools used in the tech transfer and actually how a tech transfer office works. So the moment we receive a request, one team which has IP experts, they would do the IP assessment. They will look at the IP, what sort of IP is involved in it. They'll actually, in case they identify some, so they'll quickly file and they will help you maintain the IP. Now, parallelly, the technology transfer expert will think of technology. He'll try and assess the technology at what stage it is, and then he will also perform the evaluation. That means we are actually going to check the technology market fit. We'll also look at the market, that is there actually a market, and then we'll give a valuation. Now why this technology assessment and evaluation is important? Because ultimately, before we introduce a technology into the market, we have to give some price to the technology. Like you say, please commercialize my technology. Can you actually imagine that I would give you a technology for 10 rupees? No, I cannot do that. That is injustice to all the effort that you've put in. Or can I actually simply ask for 10 lakhs too? I cannot also do that, because that for this also again I'm required to give justification. That's the reason why we perform technology assessment and evaluation, so that we give a justified value to the technology. Now we are ready with everything at our end, and the BD team, or let's say the tech transfer professional, will actually look for the right people, the right company who can actually take your technology. So they look out for the potential licensee. Once we've identified the licensee, we'll try and understand what inventor want, what the company want. Accordingly, we will actually form the strategy that how to move ahead, and then we'll negotiate the deal and technology, we transfer the technology. So do you think is technology transfer over here? No. Technology transfer means we are committing, we are giving some rights on a paper to somebody. But as of now, the technology is not into the market. So it is also the responsibility of the innovator to make sure that technology reaches market. And now the innovator has given this responsibility to the tech transfer office, who is doing it on their behalf. So now it becomes their responsibility to monitor that, everything, whatever was defined in those agreements while in the license agreement or in any different sort of agreement while transferring, are those milestones being met? Are those commitments being fulfilled? Is the amount which was committed, let's say 10 lakhs was committed, is it being paid to the inventor on time or no? If no, then we have to actually look out for strategies to actually do that. So this is called post-transfer monitoring and revenue management. So in this entire process, from signing of the technology transfer agreement till introduction of the technology into the market or launch of the technology into the market, there are times when companies require regulatory support. There are times when a startup might require team expansion or some DSIR certification or some other funding support. So for that also, TTO, they help you connect, TTO, they guide you. So this is how a TTO works. Now we'll be talking about different modes. So one by one, we'll be taking about each mode. And it is important for all of you to understand different modes, so that you can also, choose that what is right for your technology when you speak with somebody. You should know when you have developed something, then how to commercialize it. That is very, very important to have that understanding. So different modes are either we can give a license of the patent or the intellectual property that we have, or we can license the technology that we have in case it is not IP protected. Now, another is we can have a spin-off formation. We can actually form a spin-off. We can actually have a startup. The faculty, he can take approval from the university. Because see, whatever a faculty and the student they'll be developing, the applicant remains the university. That's how the policy works, the IP policy. So when they're forming a spin-off, so they will have to get the license for that technology from the university, right? Despite being the inventor. So there are times when it is being done that the university will give a license to you for manufacturing to your company, that you will have to register a company when you form a spin-off. Then another is intellectual property assignment. In license, we are either giving right to manufacture, right to sell, right to import, right to, any of these, right, or in combination. But when I say intellectual property assignment, that means it is like a buy and sell thing. You are giving, you are selling your technology in return of some money, and then you have no right on that particular technology. That means you have given all the rights to the company. Now, whatever company wants to do, they can do it. Co-development, when two entities, they come together for development of a particular product or for a particular phase of a product. So that is co-development. Joint venture, when two entities, they come together, form a third entity for a particular purpose, that is joint venture. Different tools that I say that we use, confidentiality agreement. This agreement is very important and a must for all. Whenever you share your idea with somebody, make sure you have executed the confidentiality agreement. The reason, because you never know that, how the other person can misuse or replicate the idea. Even after executing confidentiality agreement, you should be very particular about how much to share. Then material transfer agreement. In case you require some material for completing your study, or in case somebody else require your formulation for completing a study or for testing, so you should not be given free. You should free in the sense it should not be given just like that. There has to be proper material transfer agreement, and all the requirements should be properly specified, that how much material is required at what time, how frequently, and that way. Then product evaluation, in case, let's say we want to transfer a compound to somebody and the company says that, first I would like to understand the potential of the compound and then only I will seek the license agreement from you. So for that, we'll say fair enough, in case you want to test the product, whatever we are saying, you want to recheck it. So we can execute a product evaluation agreement. And for that, again, you have to be very particular that within how many days the person should submit the report and how much product will be given in case it is being shipped abroad, who is going to pay for it. So all these things have to be taken care of. License agreement, as I mentioned, again, the right to license, sell, manufacture, import, wherein one party who is the owner of the technology gives right to the other party for any of these uses. And in return, they get some consideration. Contract research, when we are actually taking up a contract research, sponsor research, when it is being sponsored, consultancy agreement, when we are seeking the expertise from somebody. So we are actually hiring a consultant on hourly basis, or on days basis. So that depends upon your university policy. Then IP assignment, in case we are transferring the IP. So different tools that we use. Now, what is the criteria for deciding that, which route to take? It is again the interest of the inventor. He wants to have a spin-off, or the inventor wants to just give it away to a large company, small company, MSME. Interest of the inventor, what he wants to do with the technology. Stage of the technology. You should always understand that how mature your technology is before giving it to somebody, so that we can actually justify the value that we are quoting for the technology, and we know that how much more investment is required in developing the technology. Nature of the technology. To which domain does the technology belong to? That is also very important to understand, because domain-wise, the requirement of a technology development changes. Potential of the technology, it's a platform technology, or it can be adopted into the existing product line by the company, or we require some new manufacturing setup for the technology. So accordingly, we have to see. Investment involved in the technology. What is the investment that is still involved? Do we need to seek regulatory approval? So this you can understand from the stage of the technology and the nature of the technology. Need of the technology. What is the market demand of the technology? According to that, in case there is very little market demand, so do you think there is a point of having a startup specifically for the technology? No. In case there is a huge market demand, so and let's say in COVID times, now if you say that I would like to set up a startup, so it might take 3 months, 4 months. By then the demand might actually reduce, or it might be fulfilled by some other competitor. So better you give it to somebody who is already good at manufacturing, who has everything set up. You simply have to give the technology, and you accordingly seek the revenue accordingly. That I'm giving you, there's huge demand, and in return I want this much amount. So that's how we can do. So let's take each one of them individually. License, beautifully defined by Boston University. They say trait. Why? It has technology rights involved in it. It has risk involved in risk assessment involved into it. And people negotiate license, so art of deal making, and we have financial modalities. We actually see that how and in what ways we want to take the revenue. A contract between parties to allow one party the right to use, sell, manufacture, or import that is owned or controlled by another party. The controlling party receives some sort of financial consideration for granting this right. Now that license could be of 3 types. Exclusive, the owner of the IP can grant only. When we say we have given exclusive license, that means we have given the right to one company exclusively, and we ourselves cannot use it, and we also cannot give it to anybody else now. Non-exclusive, the owner of the IP can grant more than one license, and and it himself also can use the IP. So the owner of the IP can grant, again, similar to exclusive, only to one licensee, but he also, keeps the room to be utilized by himself, the freedom to be utilized. Licensing strategies, we can actually license to third party, to a startup, or we can license to a large company. And another is that, you form a faculty student entrepreneur, actually you create an institute-owned venture. That means you have a spin-off, and the technology is being licensed to the spin-off. Licensing strategies, this is very important to be understood, which is being, readily followed by companies. In licensing, when we are in licensing is when company is not able to actually manufacture or can actually have that lead molecule generated of their own, because they don't have time to put in so much of research. So they would like to obtain or procure the technology from some institute, either to venture into a particular area, or to add value to the existing business. If that's a new area and they say that along with cancer, we can also sell the drugs for, let's say, TB. So they would like to enter into a particular area and they'll take the particular drug from the institute. In case it is already, it is again a cancer related product, so that is going to add value to the existing business. In this case, what happens? What all is required when we do in licensing? We will have to actually look out for a right technology. So we call that technology scouting, because we have to see that which university, which professor, which institute has developed something of this sort. So we'll actually have to look into the market and see that who has developed it. Before taking in licensing, we actually have to see whether it is worth investing or no. That we see that we can actually understand by technology evaluation when we look at the market fit for the technology, the stage of the technology, and then technology evaluation. In case yes, we are ready to take the technology, but then how much buffer do we have that we can actually, for how much can we afford to take the technology? So if it's a startup, so they have to be more cautious because they have a lot of investors who have invested into that startup, so they have a lot of equity sharing. But in case of a large company, they also have to be very clear on the terms that how much they should pay. Out licensing, it's from the university perspective or institute perspective. Herein we have developed something in our laboratory and now we are giving away the selling, manufacturing, marketing right to some other entity, to a company. For this, we have to identify a company, that means scout for potential partner. We again have to understand the technology, maturity of the technology, so that we can give right pricing to the technology and we know that what is the right worth of the technology for which we should sell. And then we have to strategize what is the best mode of transfer for us. License agreement, when we are actually drafting a license agreement, there are a few things that we should actually, these are a few major components of license agreement, I would say. Now, what is it that is being licensed? Technology or form of some IP. Very clearly, it should be defined. Define the extent of use. What is it? Is it for manufacturing or is it for selling or distribution? So we have to actually be or marketing, we have to be very clear on those terms. Define the duration for 1 year, for 3 years, for 10 years, for 5 years. We have to define it. Define the territory. Are we giving it only for the Indian territory or even the global territory or some other European country? So you have to define field of use. Let's say we have one candidate molecule, it has multiple, now we have a drug, sorry, it has, a candidate molecule, it has multiple applications. But we know that this particular company works only in the cancer field. So we'll only give it to them, give it for the use of cancer, not the other. Let's give it to other companies which have use in other fields. So that way we can actually strategize the best and we can actually have a lot of revenue generated for our university and ourselves. Revenue models. Usually what are the payment terms? So it is said that one is license fee. That, when a technology is transferred, when we give a license, so we say we want an upfront one-time license fee, which is non-refundable and it is also non-adjustable with any other form. So they'll give us, let's say, 5 lakhs upfront, and then we give the technology to them. And along with, it could be combined with any other payment mode, like milestone based. This is usually, applicable for SME and startup, wherein they don't have a very high upfront paying capacity. They cannot pay a huge amount right in one go. So we say, let's divide into different milestones. The moment you seek the regulatory approval, we will give you this much. The moment you clear the approval for the setting up a manufacturing plant, you will have to pay us this much. The moment you launch the product into the market, you will have to pay us this much. And first installment at the time of signing the agreement. So depending on the technology, we make different milestones and then we ask them to pay us some amount in different milestones. Then royalty. Royalty is something that, once we have transferred the technology and now let's sit back and let's enjoy the fruits of what we have actually developed, or let's reap the fruits. So reap the benefit of fruits. So how do we do that? Either you say that let's fix a percentage on the net sales. Whatever you will be making, whatever you will be selling into the market, you will give us some percentage of that. That's one way of doing it. Otherwise, you can fix the amount that, let's say you sell 10 units and you're going to give 10, you are going to pay us 10,000 per unit. So accordingly, different models of it. So royalty is basically once you have transferred the technology and the benefit that the company will be making, you will be getting your share from that benefit. So that is what we say is royalty. Moving ahead, spin-off. Spin-off, as I mentioned, that who can actually and when it is advisable to have a spin-off. So let's understand practically any technology which has competitive advantages. which has a attractive marketplace. They have effective IP with them. They do not have any major impediments in form of funds, and they have compelling value proposition. They have creative and realistic business development strategy and effective business model. That means you should have those entrepreneurial traits with you. You should, along with a good and strong technology. So, and a quality entrepreneurial team, which is, ready to innovate, re-innovate, and introduce new and more products into the market. So then that forms the basis of having a startup, of developing a spin-off. So it is very important for all the faculty to understand what they have in their inventories and how better they can commercialize it. Spin-off, the advantages are when you actually form a spin-off, so you become the owner of the future inventions, whatever will be coming out from the startup. Then when you actually take the license from the university, so they would ask for some equity, some share, but they will not ask you to pay huge upfront fee, or you might even get relaxed to pay milestone payment, because they understand you don't have a good funding repository with you. And then even Government of India has relaxed the patent expenses for startups. Then disadvantages, again, they have limited resources. There could be potential conflict of interest or commitment by different team members, and then deferred financial return. What you thought you might get in first year, second year, you might not get that. Assignment, it is the sale or transfer of IP ownership from an owner. This is another type of tech transfer form. It's a sale or transfer of IP ownership from an owner to the purchaser. It can be arranged by an agreement or a deed, and we can transfer present or future rights in IP in exchange for a lump sum payment. Again, it is irrevocable and permanent. Once it is done, you cannot actually redo it. Co-development, a collaboration between two parties for development of a technology. Now, they have actually, they play a joint role in addressing the problem. They put in joint contribution in the development, in the project developments. They pool in their resources. They share their benefits. They draft, mutually agreed fund utilization and IP governance guidelines, that once it is developed, that who is going to own the IP and how much, or is it going to be owned jointly? So, and then let's say we commercialize it, then who should be getting how much share? So these things have to be actually specified right in the beginning. Then joint venture, when two or more companies form a new new entity to perform a business project or activity together for a set period of time. It is time limited, that for this particular time we are because when I say joint venture, usually two companies, they come together and they form. They both have their own own businesses with them, but now they are forming a new entity. So they both look out for the profits. So they actually work together in harmony. Now, advantages, they help in formulating new market or new product line. They have access to increased resources. Of course, both of them are pooling their resources, improved expertise and technology. Sharing of losses and expenses reduced. Now, one person do not have to bear all the risk. It is being divided. Increased credibility in the targeted market with support in future acquisition. Now, differences but the disadvantage could be, now two entities are coming together. They can have differences in their culture and management style. Individual partner may not treat the joint venture as their integral part, means as their prime focus, and thus might not give the required attention. And there can be an imbalance in the level of expertise which is invested, or the investment which is being put in by partners. So accordingly, then there could be some conflicts. All right, we are towards our last few slides. It's the second last, I think. So points to consider. Now, this is for all the academia, for all the faculties here. When we execute a confidentiality agreement, so even after execution of the confidentiality agreement, it is always suggested that you should be very particular about how much information to be shared. You cannot give away all the IP involved in the into your technology. What if because it is a very contradicting thing, not a contradicting, it is actually debatable part, that how much should be shared. So it is always advisable that you should only share the information that can be published, and that in using that information, that your technology cannot be reproduced, right? So just hide the USP of your technology. Then product evaluation agreement, purpose should be clearly specified, that for this particular purpose, your product is being given to them, and not for sale, not for any other purpose. not for replicating. So these things have to be clearly mentioned for how long and when they are supposed to submit the evaluation report and their decision upon moving ahead with the technology or not moving ahead with the technology. License agreement, again, out licensing, as I mentioned, giving away the rights of marketing, manufacturing, useful for both if you have a spin-off or you are a academia. In licensing is a good option when you're forming spin-off or to be or by the startups. IP assignment agreement. IP assignment could be done from inventor to startup, university to startup, or from university to inventor. Now, there's one thing that we should always be very cautious about when we actually enter joint ownership. Then I would ask you that should it be, irrespective of the contribution, or should there be a clause on deciding ownership once the invention is ready, and then we can see who has contributed how much. So it is always advisable to keep it open, keep it negotiable, that once the product will be ready, we'll see that who owns how much IP depending upon how much effort you have put in. Again, when we talk about licensing aspects, so should there be a royalty-free license? Let's say a company is collaborating with a university and company says, fair enough, we will put in a lot of efforts, investment, and once the product is ready, you will give us a royalty-free license. Means whatever we'll be selling later, you will not get any profit from that. No share from that. So do you think, is it like, it is definitely a binding clause for the company that this is the only company to whom we are going to give the manufacturing. So it's like profitable for them. But do you think the university is going to gain anything from it? Or should there be an option to negotiate later depending upon the technology developed? That yes, technology has been developed. It is very mature technology. We have wonderful market. We have huge market for it. And accordingly, we cannot give you royalty-free license. If you want, so pay us 15% of the royalty from whatever sales you'll be making, and then we will be giving to you. So it is always advisable to keep these options open and negotiable. Now, actions that I would request all the academia to do it. I personally say that, take right step at right time with right resources. What does it mean? That, academia is the right person who understand the technology and who know what is required in its developmental phase. So when you realize that the technology is not going to work, if I carry on this particular method or path, then please stop, take a step back, improvise it, and then move ahead. There is no point wasting resources, your time, your energy. So set milestones for your research, because that helps you actually divide the resources and plan for next resources that you will be requiring for your further study. Maintain a logbook and vigilantly scout for IP in your research and report the moment you identify. You even feel that there could be a possibility of having an IP, report to your associated IP office and IP cell, and I think they should be able to help you. Validate involved factors and risk at each stage of translation. Let's say you should never be able to run out of funds or the expertise. Let's say your team members or the PhD student, they leave in the middle of the process. So you should be prepared with the the plan for how to move ahead. And follow certified, or let's say you miss out or you run out on the reagent. So plan it accordingly. Follow certified standard guidelines, standard operating procedure, and other required steps for conducting research in respective field to avoid regulatory rejection while commercialization and any unnecessary delay. So do that right from the beginning. Look for similar development, studies for advancement. Look out that who has developed similar products, attend conferences, speak or have good networking, understand from people that, do we have something similar in the market? Are we actually wasting our efforts reinventing the wheel? Then networking with relevant industry partner for co-development. Now we understand tech transfer and commercialization is a multi-stakeholder process. You never know when you require, whose support you require at which stage. So I always advise all the inventors, all the faculties to be updated with your field, to have good networking within your field, within your domain. You should know who are the different companies who are manufacturing this, so that you can actually contact them. You can definitely reach out to a tech transfer office and say, I want to transfer. Technology transfer office will help you transfer using all the tools and good strategies. But in case you already have a good relation with a company, then it becomes easy, and the hand holding also becomes very comfortable to launch a product into the market. So that's a success strategy for introducing a product into the market. Then lies with your IP and TT office all the time. Whenever you feel there is a need, you require some strategy support, you require some guidance, just feel free to reach out to them. They should be there to help you. Innovation Tech Transfer Office understand it's a multifaceted process. We extend support at every stage of university-generated technology lifecycle, and we help you strategize your win. So in case you have any queries, please feel free to write to us at queries.itto@outlook.com. And now also you're free to ask me anything. I'm here and the session is open for the discussion. Thank you.
Thank you, Dr. Dikshit, for such a stimulating presentation. You have shared much needed information actually. As if I see our university, we are having approximately 200 research scholars, including masters and PhD program. So it is going to be very important for them. Whenever you are right that so many innovations and so much research work is going on, and even I was analyzing the patent filing is somewhat increasing in our country. But when we talk about technology transfer, it again, most of the products either they are not able to enter into the technology transfer stage or they get failed. So there are
Right.
many, there are many issues. So it is good to get this presentation. Thank you. Dr. Lata ma'am, please up to you. And participants, they can ask the questions.
Participant, you can write your question in the chat box. Ma'am, Tripta ma'am, you have a very vast knowledge and you are speaking continuously. Participants were writing, you are very speed. Please, talk slowly.
I'm so sorry. I just read the chat box that, I should have gone a little slower. I apologize. In case still you have some doubts, please feel free to write back to me and then I'll be responding. I'll be happy to respond to each one of you individually. Not an issue for me. If there's anything that you want me to repeat now, I can also do that. I think we have some time left, few minutes. I can do that in case there's this particular slide that you want me to repeat.
By nature, you are speaking very fast. That's why they couldn't pick up. That's it. Otherwise, you have very vast knowledge also. Participants, if you have any doubt, you unmute yourself and you can ask questions. Ma'am is ready to answer. Rima ma'am is also waiting. If you have any doubt, you can ask.
Ma'am, there is a remark that I would like to actually make. I should say a comment. Again, this is how I started my session today. The most successful licenses that we have seen are from pharma, and trust me, what all you are working upon can actually generate a lot of revenue for your university, for the country. So go back and look into your inventories that in current situation, what can be used, because most of the universities abroad, they are doing this, that, initially they thought that, this particular drug or this particular formulation is not, useful right now. But given the COVID situation or the changing scenario, I think the requirements are changing, the market needs are changing, the needs of the health needs are changing. So accordingly, please look into your inventories and see with whom you can collaborate, how better you can generate revenue.
Yeah, right.
So there is a question from Parth Sharma. I wanted to know, wouldn't the International Patents Office, which could smoothly coordinate our work, and we would need to apply in all countries which the company is interested in applying for the patent? So Rima, you want me to answer or would you like to go? You're on mute, Rima.
Am I audible now?
Yeah, please.
Yeah.
Okay, Mr. Sharma, your question is regarding international patent application, if I understood correctly. Okay, if you want to file, it depends upon, your question is, I wanted to know, would not the International Patent Office, which could smoothly coordinate our work and we would need to apply in all countries. Okay, then I would say you have to take advice or take the assistance from any IP professional in India itself, because in India, who are IP professional, they have contacts in attorneys in international level as well, because it is always recommended to file through the attorney of those country itself, because those attorney knows the laws the IP laws of that country in a better manner. So instead of filing directly to the other countries, it is advisable to take assistance from IP professional in India first. Itself, file your patent application in India first, then decide the market potential of your technologies in other countries, and then you can file your patent application through PCT or through Paris Convention. PCT is better because you will get enough time to assess your technology itself. And indeed, the patent office in other countries will also help you. They will send notification and everything if you file through their patent attorneys, and they will send them to the patent attorney through which you have filed your application, and they will contact you. So in this manner, they will coordinate with you indirectly.
So I would like to take up the second part of the question, which says we would need to apply in all countries which the company is interested in applying for the patent. Now, in this case, if you are already collaborating with a company or you have already transferred the technology to a company, then why would you want to pay for that? So that's again because applying to different countries gets expensive. So when you are drafting such a license or when you are transferring the technology to the company and they say that eventually we would also like to seek the patent protection in different countries, so there you should have some clause which divide the fees, or which actually says that the company will pay for it. So you have to actually see accordingly, because it is an expensive process. If company says today we want to venture into Europe, so you go and apply for the Europe. Of course you can do that, but then there should be a cost sharing for that.
Okay, Miss Neha had sent me one private message. Her question was, can there be two patent owner if someone has patented a product in one country but not in other, and in that period of time someone else files a patent application in another country? Does it happen? What happens in that situation? Okay, so answer to my question is, if there is already a patent in one country, it will be treated as prior art across the globe, even if the owner has not filed a patent in that country. So there cannot be another same patent in another country if a patented invention already exists, because that must be filed after the publication of this patent. But in case of a product launch, without a patent can happen, as there is no patent in that country. So one can launch a product but cannot obtain IPR rights. And other way around, if you have filed patent application through PCT convention, right? And you want to come in India, the applicant names which you have mentioned in PCT application, then you can't drop out the name of that another applicant without the consent of that applicant. There should be mutual consent between both the applicant for dropping out the name of one of the applicant. So hope I answered your question, Ms. Neha. Yeah. In case of inventors as well, Neha, I come across one case study in which there were two inventors, and both worked together on one technology. And after that, the one inventor, he worked on that technology and further developed the technology, improvised that technology, and then he filed another patent application, which was the improvisation of the already existing patent application. But in that case also, he had to take permission from the second inventor, because the second technology itself was the improvisation of first technology. So the mutual consent is required in that case. Thank you, Neha, for this question. Thank you.
Participant, any other question?
And if any participant is having any query, they can directly contact with the speakers. They have already shared their email IDs with you all. So it was very nice to get
May
the
I
information.
just raise a query, Dr. Geeta?
Yeah.
I just wanted to raise a
Yeah.
Dr. Tripta and Dr. Reema, first of all, let me just congratulate you on an extensive, informative, and highly relevant talks. And I just wanted to know, I'm sure that the afternoon session will also be taking up topics which will be more relevant to us. I just wanted to raise a question here. This is from my personal experience where I'm just facing an issue where I want to know that we are focusing a lot of research on medicinal plants. And though we are being encouraged by the government of India to use go local and use our traditional knowledge and validate it, I find that it is highly difficult to generate a competitive edge in this kind of research. So how do you develop products or add value to your products which are coming from traditional knowledge? So do you have any experience in that kind of area where you can generate an advantage?
Rima, you wanna go first?
You please.
Okay, actually, I've come across such a request from the startup, wherein the startup says that, we have developed herbal-based products and we would like to commercialize them. So what I have understood after working with them is that, when you're solely working on herbal products, because most of the herbs, they are being recorded under TKDA. Right. So then the scope of novelty or inventive step or having or adding more value to your technology reduces somehow. But in case you can incorporate some nano composition in it, some nano formulations with them. This is what is the current trend that I've come across. Just again, you have to try and collaborate. Just try and collaborate with some nanomedicine person, and they can actually help you, combine these two studies, the herbs along with the nano composition, and then the kind of efficacy that is being generated. So you get benefited from different angles. You are more eligible towards filing the IP, and you also have like if you conduct validation studies appropriately, then you also have an edge above the existing products, because you say it is increasing the synergistic effect or it is giving a better health-wise, results. So this is what I have seen from my experience. I hope that helps.
Yeah. So that helps a lot. So then when we go for these medicinal products, you say that you develop a formulation and go for it. And then does it then still remain traditional knowledge? How do you deal with that product? Then that product is a healthcare product. You go through the official channel, or do you go through the Ayush, which says it can become an OTC? So it doesn't become an, it loses its identity as a traditional product, isn't it? Once you make a formulation out of it, how do you?
Right, to some extent that is still debatable, because Ayush is also trying to fit into the system wherein they can actually see that what are the formulation that can be qualified under our provision. And then CDSCO also, or the pharmaceutical regulatory also, is debating that, since the herbal products were used, but then now because we have added few more components and the composition has changed and also the results are changed, so it might not fall under that. So there have been times, let's say we were developing this veterinary kit. So what usually these regulatory bodies, they are actually adopting these days. They would ask you to take NOC from veterinary, Ministry of Dairy Farming and means veterinary ministry, which is dealing with them. They will also ask you to take a NOC from CDSCO. So in a way, both of them, they are satisfied. So this is something which is still debatable, because the establishment of Ayush is very recent, and the way that they work is, they say that you refer to pharmacopoeia, you refer to the TKDL, and then accordingly you come up with you can apply it to us. So they have different guidelines. So this is, again, I don't have a clear answer for it, that wherein it will fall, but then they can guide you to the best, that you take NOC from them and you also take NOC from us before you proceed ahead.
Okay, so that really is helpful, because it answers a lot of questions. But nonetheless, of course, we will we will go in this direction where we do take NOCs and help from the TTOs also. And but also, madam, that now that you are in this position where you are having a platform where you can leverage, you should also take up these issues which concern.
Right, for this, what we have been doing, because every particular regulatory body, they have their own set guidelines, and as an individual it's very tough for all of us to to know all the regulatory guidelines at what point of view. So what we are doing these days, we also have this network of regulators with us, wherein if you come up with a herbal product and you really want to understand that how to proceed further with it, so we can connect you with that consultant who will guide you better, that whether does it fall into the Ayush Ministry or the Ministry of Pharmaceuticals. So accordingly, we can help you identify that and sort out the issue. But yes, I understood your point. We should also raise these issues to the government. So we also have been given the mandate to write policy, to suggest few topics for the policy. So we will definitely be taking this up. Thank you so much.
Thank you, ma'am. I would love to be associated with your organization, and it was really wonderful. It was just much more than I actually thought it would be. Hats off to you and your team, and I'm looking forward to the afternoon session with Dr. Ashutosh.
Yes, it is also going to be very wonderful. Pleasure is ours.
Okay, there is another question from Ms. Ritu Gupta. Ma'am, you said our work will remain safe if we have filed patent in one country, then why need to apply in other countries also? Okay, Ms. Gupta, I discussed in my presentation as well that patent rights are territorial in nature. For example, if you have filed patent application in India, then you can enjoy the patentee rights, right to use, manufacture, sell patented invention in India itself. Suppose you want to stop anyone who is copying your idea in US. If you have not filed patent application in US, then you can't stop that person from copying your idea. So if you want to enjoy the same rights in US, then you have to file patent application in US itself, and likewise in other countries as well, because patent rights are always territorial right in nature. That's why if any company who is interested to launch its product worldwide, then first thing what they will do, they will protect their patent application in the countries in which they want to launch their product. Hope I answered your question, Ms. Gupta.
May I now request Dr. Geeta, chairperson, and the moderator, Dr. Lata, to please do the honors and felicitate the speakers.
Yeah, sure.
Sachin, certificate, Sachin.
Dr. Dikshit, please accept our regards to you.
Thank you so much, ma'am. Our pleasure. Completely my pleasure to be here and speak with all of them. Wonderful.
Same here. It was
for having us.
Experience.
Thank you. And we really look forward to stay associated with DIPSAR team and with all the faculties who we have today here. For any confusion, any doubt, any sort of guidance, please feel free to reach out. We'll be happy.
Yeah. Thank you so much.
Thank you so much, ma'am.
Thank you, ma'am.
Yeah, Dr. Reema, this is for you. Yeah.
Thank you so much. Thank you, DIPSAR and organizing committee for giving us this opportunity. And I'm again telling everyone who are participating in this session that if you have any query, you can write to us. In fact, you can take part in our new initiative, which is Humse Pucho, and you can book your slot on any Friday with us, in which expert from IP and technology transfer will answer your queries. Thank you, everyone. It was really a wonderful interactive session. Thank you, ma'am.
Thank you, Rima ma'am.
Thank you. Thank you, Rachni ma'am and Lata ma'am.